Okay, thank you. So thank you everybody for joining this Qualia symposium. My name is Nao Tsuchiya, and we are starting on time this symposium. So today's program is, as shown in here, we have 169 people registered by this morning. And as it says, all the symposium contents will be delivered in English. And then I'm going to give just a little bit of the introduction for 5 minutes, followed by Dr. Michael Cohen's talk, followed by Ariel's on color Qualia in peripheral vision. And then we pretty much hope this session to be dynamic and also interactive. And panel discussion will follow for 25 minutes, which is probably longer than usual kind of online talk series, but we think this is a more interesting format. So let's do it. And then after some break, we will now go to the evolution of color Qualia. And first talk will be Dr. Imai, and then followed by Mark, talking about color Qualia in evolution and development. So before starting anything in scientific housekeeping announcements from myself, we will ask you, all the audience and also potential participants, speakers, to answer the exit questionnaire that we are going to send out by the Google form. And by the way, this slide that I'm showing, sure, wait a sec, I can hear my voice. Sorry about that. In the chat box, I'm going to paste this address of the Google slide to everybody, so that if you can, if you are interested in looking at this slide itself, you should be able to look at it afterwards by yourself. Yeah, now I see many people started to see it, and then coming back to this, so from the link, you can go to the answer to these questions, and we'll also come back to this at the closing. And then during the session, I ask you all to turn off your microphone. You can leave your video on, it's totally fine, to avoid noise coming in, okay? And then we also plan to share the contents on the YouTube. Right now, we are broadcasting this thing on live through YouTube, but also after the conference symposium, we might need to edit some contents. If there is any kind of content that needs to be edited, please tell us online or chat box. And you can also speak up to ask questions during the panel discussion or Q&A, but that means that you might show up in the video. So just be mindful about that. And then we will take also the questions on either Zoom chat or YouTube comments. So please feel free to do that. And the chairs will be taking the questions for you to the speakers. And then also one other announcement is that if you want to continue discussing these things, I predict that this will be very interesting kind of symposium, and then there will be lots of discussion, follow-up literature or whatever you want to discuss. And if you are such a person, then you can join our Slack that is specifically tailored for this type of the international conversation on the consciousness. And I set up 2 threads, Qualia structure and the color Qualia, in this Slack. And you are all invited. So this one, Asia Pacific Consciousness Network, you can join from this thing. Okay. Wait a sec to come back to this. And share session again. Can you see the screen right now? screen right now. All right. So then, just to also briefly introduce some of the characters that you see behind my screen or background or some of the slides. We did a mascot character contest for our grant that supports this entire symposium, Qualia structure. And these are the 3 characters plus logo mark that was selected based on the public application. This is the Qualin and Nomin chosen from 7 to 15 years old applicants. And this is a SWR from 16 to 20 years old section. And this is the Mr. Qualia or Qualia-kun, 23 to older population. We are trying to sort of raise the awareness of the term Qualia in Japan and through the world, in a sense. So just briefly, I want to also briefly mention this entire grant that is supporting this initiative. This is called the Qualia structure, granting aid for transformative research areas. And the member is including myself and Makiko Yamada and Masafumi Oizumi. And we are trying to understand the structure of the Qualia or how it's generated from or supported by brain activity by combining information theory and psychophysics and the neuroimaging and neuromodulation. And that's it from myself. And then please enjoy the symposium. And I'm going to ask Masafumi to take the chair.
Yep. So let's get started. So thank you for joining us. My name is Masafumi Oizumi from the University of Tokyo. I'll chair the first session, Color Qualia in Peripheral Vision. The first speaker today is Dr. Michael Cohen from Amherst College. The title is How Much Color Do We See in the World? Please, Dr. Cohen.
All right, so I will take over and share my screen. All right, can everybody hear me okay?
Yes.
All right, so thank you all for being here. Thank you very much for the invitation, for sure. And I'm just gonna dive right in so we can really hit the ground running. And the question that I'm going to really focus on is how much color do we see in the world? And the logic of the approach that me and my colleagues take in answering this question is actually relatively simple. So basically our idea was, we just wanted to know how much color could we remove from the world before people notice. So we're going to show people a bunch of stimuli, we're going to start systematically removing color from the world, and we're going to kind of look for the breaking point, or the point at which people start to spontaneously stop and say like, wait a minute, something's not right, you've removed some color from the external world. And the idea is that if we remove colors and people don't notice that we did that, then our inference would be that those colors that we removed were not a part of their active perceptual experience. So to do this, rather than do it like on a computer monitor, like we're generally accustomed to in psychophysics, what we actually did is we put people in a virtual environment. And we just told them to look around a bunch of natural scenes. We gave them no task whatsoever. We just told them to explore the world in whatever way they felt natural to them. We didn't give them a primary task or anything at all. We just gave them about 20 seconds to explore the world. So people would come into my lab. We would outfit them with a headset like this, just a standard Oculus virtual reality system. And they would just have the freedom to sort of naturally explore their surroundings for a little while. And while they were doing this, we would use eye tracking that we actually implanted into the headset itself to monitor where participants are looking in the scenes at all times. So from one moment to the next, we actually always know where people are looking. So what I'm going to do now is I'm actually just going to show you a video just to give you a sense of the setup before we get to the experiment itself. So this first video will just be showing you participants sort of naturally exploring a virtual world that they're embedded in.
Cassies were designed for blinders, and they brought them over from England. You know, Cassie is a mutant.
Okay, so you could just see he was naturally exploring. He didn't really have a task. He was just checking out the world around him. Now here's a video of what it looks like from his perspective, or from the inside.
My name is Chris Sussens. I'm the manager or ranger here on Chukudu Wildlife Reserve. We're in a very severe drought at the moment, so that's why we're actually feeding these.
So you could see his eyes moving down on the bottom right, and you could see that little blue dot, which was showing you where he was looking. That was one video. Let me just show you one more just so you can really get a sense of the diversity of our stimuli.
We have underway, we're making a bunch of dummies to collect shrapnel from a blast. And we also have our spray booth, or the area that we do spray painting or vent fumes. And there's the famous wall of boxes. The boxes have just about everything that you can imagine that you could ever want.
So people come into the lab, they see a bunch of different types of stimuli. I think they do, 8, 9, 10 trials. They're just exploring. And then what we do is on the critical trial, as we call it, we desaturate the periphery in this gaze contingent manner so that wherever they look is in color, but wherever they look off into the periphery is not in color, will be desaturated. And this is actually inspired by work done by Keith Rayner many years ago, even days back to the 70s. In this domain, they didn't do it with color, they did it with reading. And so, for example, you can think about there's, 3 different time points while you're reading a small sentence like this, and you can look at, across time, your saccades move and the fixation point changes. And it actually turned out that what these researchers have found is that if you replaced letters in the periphery with something just like a string of X's, that observers would actually not even recognize that they were looking at an altered text, so long as you kept those patterns there. Of course, though, if you removed the letters altogether so that it was blank, then they would very quickly notice that something was wrong. So this was really the source of our inspiration, is to create this sort of gaze contingent paradigm. But we didn't do it with reading. We obviously did it with images of natural scenes and with color. And so, for example, if a participant in the critical trial was staring at this dot, we would perhaps make this man's face remain in color while the periphery would be desaturated. Or if they were looking at this dot in this restaurant, this center part here would be in color and the periphery would be desaturated. And so what we're going to do on the critical trial is, as people are moving their eyes around, we are going to just desaturate their periphery in this gaze contingent manner. And we're going to do it with different size, as I call them, color windows. So we're going to have a couple different conditions. So to give you an intuition of what these conditions are, I want to first orient you to these 2 images here, this is intended to represent your field of view. So for example, if you were staring at this dot here and you closed your left eye, you would see this sort of line that, actually is caused by your nose from one eye and then from the other eye. So this is like the entire field of the visual world. It's about 200 degrees left and right and about 110 degrees up and down. So in our biggest condition with our largest color window, we're going to make the color window, in this case, 32.5 degrees of visible angle across. So basically what this means is this colored circle here is giving you a sense of how much of the world will remain in color. And then out here, this is the size of the world that will be in black and white. So this is our largest condition. Then we have a somewhat smaller condition, a somewhat smaller condition still. And then actually we go down to just a 10-degree circle where a very, very small percentage, as you can see, of the visual world will be in color. The entire rest of it will be in black and white. I do realize that seeing your field of view like this might be a little counterintuitive if you're not used to seeing something like that. So to give you another scale of what your field of view is, pretend this is, our participant, we're looking down upon them. This is, to the left, this is to the right. 32.5 degrees looks like that. This is a smaller condition, a smaller condition. And again, our smallest condition at 10 degrees of visual angle is very, very small. But let's start with a critical trial where we remove the peripheral color with this largest condition. So what I'm going to do now is I'm going to show you a video. And this is going to be one of these trials where a participant is just looking around their environment, and unbeknownst to them, we are going to start removing color from the periphery. They have no prior expectation this is going to happen. They think they're just doing a simple virtual reality study about scene perception. And so what we're going to do now is, without them having any prior knowledge, we're just going to remove the color from the periphery. And this is going to be the actual trial from one of our participants. So let's watch.
But this is the hobbit hole. This is the main one.
And as you can see, the color is starting to desaturate
as well, I think. See if you can have a little peek inside.
And so that color window that was moving around because that's where our participant was saccading. So the moment that this trial ended, that the screen would go to black, we would immediately ask our participants a progressively specific series of questions. So as soon as the trial went black, what we would do is we would ask them, did you notice anything strange or different about that last trial? And if they said no, or if they gave an answer that would have been like, well, seeing a hobbit hole, as that man said, is kind of odd, but that's not really what we're talking about. We would ask the next question, which would be, if I were to tell you that there was something different about that last trial, could you tell me what it was? If they say no to that, we then say to them, if I were to tell you that there was something different about the very last image you saw in the last trial, could you tell me what it was? Trying to kind of draw their attention to what we were doing near the second half of the trial. And if they said no to all these different questions, we would actually put them back in the altered world, and we would show them the effect to confirm that they didn't notice. So we would put them back in there, and we would even say to them, okay, well, what we did is we actually altered the periphery, so please pay attention to the periphery. Now can you see that, we're removing color, and so on and so forth. So we would ask them all these series of questions, and what I'm going to do now is I'm going to plot on the vertical axis the percentage of participants who would claim that they didn't notice the peripheral desaturation, but I'm only going to plot the participants who said no to everything. So these are going to be participants who, when I put them back in the virtual world, will say something to the effect of, I do now see that the periphery is black and white, but no, I did not notice that a moment ago. So it's people who really will see the effect and actively deny it. And so down here, I'm showing you the size of all the different colored regions. We just saw this display size over here with 32.5 degrees. And in that case, a good majority of our participants, 83% of them, were completely oblivious to the fact that a good portion of their visual world would be completely desaturated. So that's what the effect looks like at 32.5 degrees. Let's go down to 25 degrees of visual angle, and let's watch this video.
Howdy, I'm Brian. I'm Justin. And we're checking out the best parts of Austin here with Discovery VR. And when they say keep Austin weird, I'm 99% certain they mean this place.
We are in suburbia, family-friendly home. And then we pull in here to see one of the most amazing structures I've ever born with.
All right, so in that situation, when you remove that much color, 65% of people, so about 2/3 of people, are still oblivious. They still deny that they still claim that they didn't notice the desaturations in the periphery at all. We can go down a level still to 17.5 degrees.
I wanted to show you guys what's new this year. This year's focus was to try to get more product through our plant. We knew to get to 2,700 ounces, we had to run this thing pretty hard. And our biggest problem last year was our tailings. If you look way down there, you see big piles of gray rock. That was all of our tailings. So it took a lot of...
All right, and in that situation now, 50% of people don't notice. So we're starting to see that as that colored window gets smaller, the rate at which people are inattentionally blind or not noticing the desaturation does in fact drop. But it's still a significant, a good portion of people are unaware of it when we remove a tremendous amount of color. Now I'm going to show you the very smallest one at 10 degrees of visual angle. And to give you an intuition of really what this looks like, 10 degrees of visual angle in this particular condition. Oh, sorry, I didn't realize I changed slides. 10 degrees of visual angle corresponds to about 7.5 inches at arm's length, or about 10 centimeters at arm's length. So it's a really small portion of your visual field. And so let's see what that video looks like. Oh, sorry, here we go.
What is a highlight, but we don't even know what it is? What is a highlight? Highlight is the world's fastest sport. How fast does it go? 180 miles. That is fast. All right, how do I fling the ball so it goes that fast? All you have to do is keep your wrist loose all the time and throw the ball to the front wall. So it's similar to racquetball or squash? Yeah, kind of, but fast.
So even with something as small as 10 degrees of visual angle, 30% of people, close to 1/3 of people afterwards would be put back in that video and would just be completely oblivious to the fact and totally denied that they did not notice that the color was removed from their periphery. And I really just want to stop and emphasize, like 30%, 1 out of 3 people don't notice when we remove that much color. To me, I found this very shocking because it's, again, such a small amount of the world that remains in color. So to give you a bigger sense of maybe why I find that surprising, let me just show you one more video from this condition. And again, this is actually a video of, you were going to see what a participant saw who later claimed that they didn't notice it. So this is actually an example of a participant who did not notice the peripheral desaturation.
I've got the gold all dried up. And we're going to find out here in just a moment. There you go. That's 20. There's 30. Here's 40.
So very, very little color remains in the field, and yet about 1 in 3 people don't notice. And I really just want to make sure to emphasize that in this case, there was no tasks whatsoever. It was as close to real world viewing as possible. We really did that in purpose because we wanted people to allocate their attention in a very natural setting. We didn't want to make any assumptions of what natural attention is like. So we purposely gave people no instructions, and we wanted them to just look at the world naturally. But of course, a natural question to wonder in light of these results is, well, what if people do distribute their attention and are looking for the desaturation? Kind of, what are the outer limits of color awareness if you instruct people to look for those peripheral desaturations? So to answer this question, what we did is this thresholding experiment where we would want to find the point at which participants were basically at 50-50% chance, like, basically at chance at saying yes or no, did we desaturate the periphery? So what we would do is we would put people back in the world. At some point, we would remove color from the periphery. We would ask them to press a button just to say, yes, did they remove color? No, did we not? And we just wanted to know how big could we make that color window while people would remain at chance. And when I show, what I'm going to do now is I'm going to show you this for 20 individual participants, and you get something that looks kind of like this, which on average corresponds to about 37.5 degrees. If you're anything like me, 37.5 degrees, you might not necessarily know what to do with that number. So what I'm going to do now is I'm going to plot this number of the 50-50% threshold, again, in this sort of field of view data. And in this situation, it was about this big, that if we made the colored region about this percentage of the visual field, people were around 50-50. If we made it any smaller, they'd start to notice, but if we made it any bigger, they would not really be much better. And to give you another intuition of how big that is relative to a person, it looks something like this. And this is bigger than the previous 4 conditions that I had. These are the 4 conditions I had from the inattentional blindness condition. And so sure enough, there is an attentional effect that once you tell people to kind of widen their view, they can definitely notice more color than in these sort of situations where their attention was presumably a little bit narrow. So overall, I think what we'd want to argue is that in everyday life, your visual experience might be shockingly less colorful than it intuitively seems, and that in fact we can remove a tremendous amount of color from your visual world and you actually don't even notice. To me, I think the most interesting question, and I think really the root of where a lot of debate may be, is why doesn't it seem that way? I personally don't have the impression that I see that little color. I get the impression that I see much more color than my data suggests. So why doesn't it seem that way? And this is not a trivial question. In fact, people have been asking questions like this since, change blindness and inattentional blindness were first discovered. And so this is really at the forefront of our interest right now. And so let me just tell you real quick about a few current and future directions we're working on right now. So one thing that we thought about why people may not necessarily feel as if they see so little color is that in everyday life, we're not in novel, brand new virtual scenes. We're in very familiar scenes. I'm currently sitting in my office. You're at home. You're on your, at your child's school. You're in places that are very familiar. And so perhaps there's going to be significant differences in what your color thresholds are going to be for familiar versus unfamiliar scenes. And perhaps there will be different rates of noticing when it's a scene you know very well. So we actually are running an experiment where we have filmed scenes in very highly familiar locations to our participants and comparing their color threshold to unfamiliar locations. Similarly, we've been making some efforts to try to find more direct and explicit evidence of perceptual filling in, that maybe what's going on is that people aren't noticing the lack of colors in the periphery because something like memory is a process that is filling in those peripheral colors. So people are still perhaps having the subjective experience because their brain can fill in the green of the grass or the blue of the sky and things of that nature. Oh, sorry. My slides just got a little messed up. Oh, Christ. Oh, sorry. Well, the last future direction we will skip for right now because my slide seems to have had a problem. But I will stop there actually and happily take times for questions. I do want to just alert everyone that if people are curious, this paper came out a few months ago in PNAS, so please check it out. And I'd like to quickly thank my 2 collaborators on this work, Caroline Robertson and Tommy Botch, who are both at Dartmouth. Caroline is a professor there. She has a lab where they do a lot of work with VR, so I was really able to work on their infrastructure to do a project like this. And thanks to you all so much, and I'd love to take your questions or hear your comments or your thoughts. All right, thank you.
Yep. Thank you for the wonderful talk. So we now take the questions from the audience and the panelists.
Anybody any question? Yeah.
Can I ask a question?
Yes.
Or do I need to raise hand?
Yeah.
Yes. So, hi, Michael. Very good speech. I just have a question. When you do the VR experiments, do the trials always start with full color scenes, meaning that the entire scene is full of color and then it gradually decays?
Yes, that is right. The trial would always start with full color. I don't really know what would happen if we started out, with the effect immediately going on there. I think one of our concerns was that we were worried that right when the scene comes up, we didn't have a real good sense of where people's attention would be. Maybe it starts broader as they're first getting a sense of where they are because it's going from one location in the world to another. So yeah, the first, I believe it was about 7 seconds, would always start in color. And then over the course of a couple seconds, it would fade out, and then it would remain fade out for about the last 10 seconds or so.
Oh, okay. Do you think it's possible that some kind of prediction or expectation about that the periphery are going to be colored at work?
In a sense, I actually really do. I actually think that this is now me just speculating. I think there's a really good possibility that what's going on is that we all have these really strong priors that the world is in color. Like that's an embedded prior from our entire lives. Like every moment that we're conscious, we see the world in color. Yeah. And so to overwrite that prior and to overwrite and update that belief to think like, oh no, I need to notice that there's something strange going on in the world. It has to be a really, really, really strong signal that is so powerful that we're like, wait a minute, this thing that I usually take for granted, which is a colored visual world, I can no longer take that for granted. And so I think, though, that it becomes this distinction we need to make between people knowing that the world is in color and assuming the world is in color and having the belief that the world is in color versus them actively in the moment perceiving that it is in color. And so I think there is this interesting tension about how do you reconcile the fact that people have these prior beliefs that are very well entrenched in their mindset, and how are they going to get influenced by the current input of data that's coming? So I certainly think there's a big role playing with that right there, just like you mentioned.
Yeah. Okay. Thank you.
Sure.
Thank you. So the next, Nishida-san, can you speak?
Yes, thank you very much for nice talk. And my question is about if you evaluate the size of the visual area, that you lose the sense of color, probably that's very huge, but you can evaluate it in terms of a proportion of cones or a proportion of ganglion cells, color selective one. And have you ever tried to evaluate it in those terms?
We've made some initial efforts because some people have asked us, like, is it possible that what's going on is this is entirely like a retinal bottleneck? Especially with like the very small 10 degrees of visual angle, like maybe for some people, they just don't have that acuity in the periphery. That's not exactly the case because the data, when we had people distribute their attention to the periphery, it was much wider than the fovea. And in fact, as you know, a bunch of people here might know, now has some work with Andrew Hown and Christoph Koch, where they show that if you scale the items large enough in the periphery, you can still see some color, and that there is a scaling factor that plays a role in it. So we haven't done an exact sort of one-to-one matching besides trying to see like, oh, is this potentially just caused by the distribution of cones on the retina and specifically in the fovea. So I think there is something going on cortically, even attentionally, or even this higher belief system over and above whatever the limitations might be that get imposed from the retina itself.
Yeah, I generally agree with the argument, but I just want to see what happens if you see the similar proportion of the cones are lost in the central visual field, or similar proportion of that kind of demonstration will give us a more clear idea on what's the critical difference between the peripheral and central. So
yeah, I think I agree with you. I'd actually love to know if there's a way that we could directly quantify like what exactly is the input from the eye to the brain, like when the rubber hits the road and when like things first get started in terms of perceptual processing, there's already going to be limits imposed by what the retina can capture, and I'd love to be able to visualize and quantify what those are. We just, to be honest, haven't been able to do that at this point.
Thank you.
The next one is Sunny Lee. can you speak?
Yep. Hi. Thanks, Dr. Cohen, for the very interesting talk. I was wondering if you've tested or are considering testing how long it takes for people to notice that this desaturation has occurred, because with these VR scenes, they're very novel and you're just suddenly throwing them into it. And to be frank, for a lot of people, VR is a cool new thing. So they're just very interested in exploring and not really paying attention to things. So I could really see this as there's an additional attentional component where in a 20-second window, it's very easy for a lot of people not to have noticed it purely because of this very novel scene. And there's like a lot of talking going on. So they're like focusing on faces rather than exploring the environment. So I was wondering if you consider testing that.
We have actually a little. The reason it gets really tricky is that people sometimes do really different things. So we've done things where we make it like several minutes of a person in there. And for example, you could have one person, and we've had participants who do this, who after a couple minutes, they're like, I've been in this scene for a while. I've been looking around. It's not that interesting. I'm waiting for the next trial. I don't know how long I'm going to be in here. I'm thinking about homework. I'm thinking about my exam. I'm thinking about what I'm going to have for dinner. They really kind of disengage with the visual world, and they start thinking in their own head because that's what their mind's just wandering, like my mind would wander if I was on a street corner or whatever. Some people will be like really fixated. So like, say you were looking at a scene that's my office. They might be like, oh, like what are all these books over here? Like, oh, that's red. That looks like a book by, Shin Shimojo or something over there. Maybe Dan Dennett, or like, what is that? Is that a stuffed animal? Like, and they'll focus their attention a lot. And so we got a little discouraged in trying to figure out how to manipulate this issue with time, because as soon as you change the timing of it in a substantial way, people start diverging very quickly in terms of what they're doing attentionally. And so we were getting very different results, but when we would talk to our participants, we could kind of get a sense of why, because they would very easily describe like their attentional state. And so I think it's a worthwhile question, especially as it relates to something like familiarity and memory and knowledge, but it is just difficult because there is always this factor of how much people are zooming in or zooming out with their attention.
All right, great. Thank you very much.
The next one is Sharon Daniel, please.
Yes. Hi. Thank you for the talk. I was wondering if you try to change other features on the periphery beside colors. And another question maybe, if you have tried to do a reverse experiment where everything is black and white and you change the color in the periphery.
Oh, like everything's black and white and we add color to the periphery?
Yes.
I have not done that. But to your first question, so hold that thought 1 second. I will find it. We got interrupted by the COVID pandemic with what we were doing, but we have done things where we change other features in the periphery. So let me show you some screenshots. And some people might have seen things like this from prior talks that I've done. But basically what we would do is we would have these black and white scenes, and we would start to systematically scramble the periphery as people looked around in a virtual reality setting. So this could be like a mild scrambling setting, and this would be a more significant scrambling setting, and then you can have an even more significant scrambling setting. And so we have done things that in the headset as well that are moving beyond just color and that are looking things like the statistics of the visual world, the structure of the visual world, and what the resolution of those things are. We've just unfortunately been slowed down a little bit because social distancing precluded us from getting more data. But that's something that we actively have going on right now. For your second question, no, the short answer is we haven't done the opposite of putting in color when it's black and white. But to be honest, I think there's a lot of interesting work there, again, that relates to the first question about priors and beliefs and knowledge, because I think that there could be some opportunity to do things where if you put people in a black and white world from the get-go, they're immediately going to update their prior, where they're going to think like, okay, now I'm in a virtual world that is black and white. My long-held belief of seeing the world in color has been violated. And they might really be able to notice these things much more differently at a different rate, just because their sort of prior expectations and understanding and beliefs have been radically updated. So we haven't really done that yet, but we've thought and discussed that a lot about that sort of, I think if that's where you're kind of getting at, I'm right there with you.
Can you please share with us what have you found for my first question? If people were aware of the changes?
Sorry. So we actually need to move to the next speaker. So So I
short answer is yeah, people don't notice some stuff. Yeah.
Yeah. So the next one will be the last question. So Catherine Atkins, please. Catherine, can you?
Kathleen, you're muted.
Yeah.
I know. Here we go. Sorry. I had a very different kind of question, which is, given that chromatic contrast plays sort of a complementary role to luminance contrast, that is to say, it highlights different kinds of contours, it can increase, what were the previous luminance contours and so on. Once you remove the color, you remove the chromatic contrast. So what I was wondering was, is this really about color or is it about contrast? Because these are deflated or conflated, sorry. So my question was,
sorry, go ahead.
Okay, so my question was, I noticed that in all of the films you had, they have, of course, because they're commercial films, they have very high luminance contrast. If you decrease the luminance contrast, would that make a difference to whether people noticed the color was missing?
My prediction, if I understand, I'm not 100% sure I'm confident in knowing how to visualize what it would look like to do that. But my hunch is that actually people would notice the lack of color more, or rather, to put it simply, they would not realize when we do the removal, because if the world is already not having that much contrast, it's not as saturated or, whatever, then it will be easier to remove some color and we can kind of slip under the wire and it won't be as apparent when we do remove it. So my personal prediction is that the effect would go up. Oh, really?
Yeah, the opposite, because look, you need a certain amount of contrast, any kind of contrast to see. You reduce luminance contrast, which is sort of the biggest factor. And this is when you see sort of a great increase in sort of visual acuity and seeing objects when you add the chromatic contrast. So now you remove the luminance contrast, and because now you have a poor signal, the question is, where's the signal that I used to get all that information from?
Mm-hmm.
Okay, so on this view, what sort of tips you off, you as the conscious person, is the lack of being able to see.
Right.
And then in virtue of that, you notice that the color is gone.
Yeah.
So there are two very different mechanisms here.
Yeah, that's interesting. I do wonder if perhaps what I'm picturing in my head is different than what you're actually intending, and that's where a difference in our predictions comes from. The one thing I will say that we are doing is we are trying to get a sense of what are the visual features in the world that do cause some people to notice it sometimes and not others. So not necessarily just looking at individual differences between people, but differences between the scenes themselves. And so we've been taking a sort of big data approach of getting lots and lots and lots of data and trying to use a deep net to try to isolate the features and learn and predict which scenes are people going to notice the effect, which scenes are not, and then use different sort of visualization techniques to try to zero in on which layers of a network best predict it, what features are they. So we're doing it in a more hypothesis neutral way, but I think your point about that confound, I completely agree with you.
Okay.
Yeah, thank you. So yeah, actually we now need to move to the next talk. Thank you.
Thank you.
The next speaker is Dr. Ariel Zeleznikow-Johnston from Monash University. The title is Are Color Experience Structures Equivalent Across the Visual Field? So please, Ariel.
Thanks. You can hear me all right?
Yes.
Excellent. Hi, yeah, I'm going to talk about an ongoing project to try and determine whether relationships between color experiences are the same across the visual field. So my first guess at this would be obviously yes. It certainly seems to me that my naive impression is that there's some external world, light bounces off of stuff in it into my eyes, that generates color experiences across my visual field, and then I'm able to report these color experiences to all of you. But as Dr. Cohen and many others have pointed out, there's evidence from neurophysiology and psychophysics to be pretty suspicious of this. So we know that cone density decreases dramatically outside of the couple of degrees around the center of vision, so the fovea. We know that there's pretty dramatic differences in acuity as you increase into the periphery. We know you get effects like crowding, so it becomes difficult to distinguish individual stimuli. And we know that there's increasing color contrast required in order to detect changes in color in the periphery. So there's all this evidence to suggest that maybe the naive view isn't correct. How then are we going to go about testing whether these color experiences, their structure is the same in the periphery? So one way to do this is to show people colored stimuli, which presumably generate colored experiences for them, and then get them to rate how similar these are to each other. And the idea is that if you do this systematically across all the different possible color combinations, you get a set of similarity or dissimilarity relationships. And then you might be able to define what an experience of red is as something that is identical to any other red experience, or very different from a green experience, or very similar to but not exactly the same as a pink experience. And collectively, you can use this set of similarity judgments to try and define the structure of color experience. Now, previous work has looked at these color similarity judgments in central vision, but to the best of my knowledge, nobody's done this for peripheral vision. There are researchers who've looked at how people rate the same stimuli that are shown in central vision, then presented in peripheral vision along predefined axes, such as what's the hue of this stimulus or what's the saturation of this. But we're not aware of anyone who's looked at similarity judgments with these stimuli presented in the periphery. So we want to, in this circumstance, look at the central to central comparisons again, but also central to peripheral comparisons and peripheral to peripheral comparisons. So I'm going to run you through how we did the experiment in these COVID times. So we got individual participants to do this on their computer screens at home. So we recruited participants from Amazon Mechanical Turk. And the first thing we needed to do in order for them to do this task is to get a sense of how big their screen was, where they were sitting relative to it, so that we could reliably present stimuli in particular locations with particular degrees of visual angle. So we start by getting their screen size. So we show a square on the screen. We get them to place a standard size student card or credit card on the screen, and then adjust this box so that it matches up with their card, which gives us their screen size. And then for a typical laptop screen, we'd also get them to position themselves about an arm's length away from their screen. So they'd see something like this, where if you want to follow along on your own laptop screen, you can extend your thumb out and press it to the circle. So that means we've got their screen size and their rough viewing distance. And then to get a little bit better with their viewing distance, we can also take advantage of the fact that people's blind spot is pretty much a standard degree offset from their fixation point or their center of vision. So if on their screen, you get them to fixate in a particular location, and then you show them a dot and you move it across the screen, if you can ask your participants to indicate when this dot disappears, if you ask them to indicate when the dot disappears, you can get their viewing distance using this angle as well that you can obtain. So collectively, what this means is that if you're viewing this on a laptop screen, we did have about this degree of visual angle to play with. So if they're fixating in the center of the screen, we should have about 15 degrees either side of that where we can present stimuli. So 15 degrees is a lot less than the full range of human vision, but it is significantly outside of central vision and outside of the fovea. So cone density drops off again pretty dramatically, pretty quickly within just a couple of degrees of the center. So when we were showing stimuli to our participants in central vision or in peripheral vision, we would be showing stimuli somewhere along these radii from the center. So the central stimuli at 1 degree and the peripheral stimuli at 10 degrees. And to give you an indication, this is a central trial, for example, where they'd see 2 stimuli like this, and they'd be asked to rate the similarity. This might be a peripheral example where there'd be 2 stimuli like this. And we also tried different sizes of the peripheral stimuli. So sometimes we kept them fixed relative to the centrally presented stimuli, and sometimes we had magnified or peripherally scaled stimuli, which I'll go into a bit more detail about later. We also did center to peripheral comparisons. So to give you an example of what the experiment was really like for participants, we'd initially show them the set of color stimuli that they would be presented with later in the experiment, sorry. We'd get them to, on each trial, fixate in the center of their screen. We'd show them briefly the two colored stimuli to try and prevent them from saccading to the stimuli. Then we'd get them to rate how dissimilar the colors were. So from 0, which is most similar, exactly the same colors, through to 7, which is most dissimilar, completely different colors. And we then get them to click in the center to get them to fixate again for the next trial. And just to show you another example, this time with peripheral stimuli, again, click to indicate their value, click to fixate again in the center. And the idea was that we had 10 different conditions that we had, or 10 different subject groups that saw these stimuli. So we had peripherally fixed stimuli, where the stimuli were the same size in the center and in the periphery, from 1 degree radius to 0.05 degrees radius. And we had peripherally magnified stimuli from 1 degree radius to 0.05 degree radius, but magnified in the periphery in that circumstance. So what were the actual results from this? What did participants give us the same similarity ratings for centrally presented and peripherally presented stimuli? Before I get into the summary statistics, I think it's helpful to show you a couple of individual subject response examples. So this is one subject who is reporting a fairly continuous set of dissimilarities across the colors. So they're blending neatly from red into green and then back towards red in a graded fashion. And they're doing this in the center and the periphery and center to periphery comparisons. This is a subject who's being a fair bit more categorical about their judgments. And this is a subject that's being perhaps a blend of both and perhaps has a different set of similarity judgments when they're making center to peripheral comparisons. This is only to point out that there's a pretty interesting pattern of responses you can get from participants and some degree of heterogeneity between the individual participants. So looking at the actual results. So the first question is, at a global perspective, are there just generally correlated judgments between comparisons made between colors in the center and comparisons made between colors in peripheral vision? So you can see here, first looking... sorry, is someone asking a question? Oh, okay. As you can see here, and I'm first going to present the peripherally magnified stimuli. In this circumstance, the color similarity judgments made in central to central comparisons are very, very highly correlated with the judgments made in center to peripheral comparisons or peripheral to peripheral comparisons. So this, at a high level, initially suggests that the color experience structures are equivalent across the visual field because participants' judgments are going in the same direction. We can break this down a bit and break it down by stimulus size. You can see for the peripherally magnified stimuli, the size of the stimuli themselves don't really make any difference to the degree of correlation that subjects report. So for smaller or larger stimuli, as long as they're peripherally magnified, subjects think the color experiences are the same. But if you use fixed size stimuli, you see that for the larger stimulus sizes, participants report that the color experience structures are the same. But below a certain critical threshold, these correlations start to break down, which suggests that with sufficiently small stimuli, perhaps the color experiences aren't equivalent in the periphery to the center of vision. So that's looking at the overall relationship structures, but what about the particular color comparisons? Showing a global correlation in the judgments across eccentricity doesn't necessarily mean that they're always the same. They might just be co-varying together. So do participants actually give exactly the same judgments? The way that we examine this is for any particular subject, we can take their central to central comparisons and their peripheral to peripheral comparisons, and we can compare all of the individual entries in these dissimilarity matrices. So we want to know, for example, if these two values are the same. So that's to say, for each subject, is their green to red comparison the same as their, is their green to red comparison in the center the same as their green to red comparison in the periphery, i.e., is the difference between these dissimilarity values equal to 0? Now, if we do this across all of our subjects, we can get a histogram or a distribution of all of their responses. And what we're asking is the difference distribution here tightly clustered around 0? Which would be the same as asking, do participants pretty much think that these individual values are the same? And we can do this for each subject and across all the different comparisons between central to central colors and peripheral to peripheral colors. And just we can get into the stats in more detail later if you want, but this is just to give an idea of the confidence interval of the means for these distributions. This is taking the mean across all of the different color comparisons for each of the groups. So looking on the right here at the peripherally magnified stimuli, you can see that the confidence of the mean for those groups is pretty tightly clustered around 0. So it's within about half a point of dissimilarity either side. So for these populations, it would seem that generally the subjects are saying, yes, the peripheral to peripheral color comparisons have the same dissimilarity as the center to center color comparisons. For the peripherally magnified, for the peripherally thick stimuli, so when we're using the same size stimuli in peripheral vision as in central vision, again, for smaller stimuli, the confidence that the mean is around 0 is pretty tight, but this also then expands as we get further out, as we use smaller and smaller stimuli in the periphery, meaning we're a little bit less sure that they're making exactly the same judgments in the central comparisons and the peripheral comparisons. So those are my results, but what is it that I actually want you to take away from these results, showing that these similarity structures are the same? So the primary thing is that it would seem that the structural relationships between color experiences in the periphery, at least up to the 10 degree or so limit that we've been able to test, seem to be the same for those, seem to be the same between peripheral vision and central vision. And here is just a two-dimensional, multi-dimensional scaling representation of that dissimilarity data. And the idea here is that the distances between the points reflect the degree of dissimilarity rating differences. And you can see that, for example, red is pretty distant from green on the other side of this matrix, of this graph. But the points that correspond to the central judgments and the peripheral judgments, or the central color and the peripheral color, are pretty much overlapping, indicating that participants think that they're pretty much the same. Now, how can this be the case given these, we know that there's these cone density differences across the retina, and we know that there's these other behavioral differences that we see. So one thing to point out is that with increasing eccentricity in the retina, the number of cones that are pulled together gets larger and larger. So the receptive fields of retinal ganglion cells, which pull cone information, get larger as we move further into the periphery. Then V1, which collects these retinal ganglion cells, gets larger. V2, V4. There's an increasing receptive field size corresponding to larger and larger portions of the retina as we move up the cortical hierarchy. This is just to say that there's a possibility of keeping the cone density to receptive field area roughly constant across the periphery, meaning that there should be color vision in the periphery available to areas of the brain, even when corresponding to quite peripheral stimuli. So that there's color information available in the periphery is the point. How then do we go about actually reconciling this with the other behavioral data that indicates impaired color experiences in the periphery? So my first point would be to say that It is important to keep in mind this scale invariance in magnification. It's not necessarily the case that foveal vision and peripheral vision are the same, and we can make out the same level of color detail in the periphery as opposed to in central vision. But there is also relevant color information at larger and larger scales. And this figure on the right is just to demonstrate that you can have interesting things at smaller scales and at larger scales. The second point, which Dr. Cohen already touched on, is that there are inattentional amnesia and blindness effects which can account for people's inability to notice peripheral color changes in their, peripheral color changes. But these same inattentional amnesia and blindness effects can happen in central vision, not just in peripheral vision. So there are experiments to show that if you have participants attending to their periphery, you can also alter stimuli that are presented in their central vision without them noticing the effects at all. So the problem doesn't just apply to color vision in the periphery. I'd also like to point out that there are other clear examples of dissociation between actual perceptual information or perception and the experience that individuals have. So filling in is a simple example. We all seem to experience a constant visual field, but there's an absence of perceptual information coming in over our optic, our blind spots. But more dramatic examples are the fact that we have, or at least many of us have, color experiences every night in the complete absence of, or near complete absence of perceptual information coming in. For example, during dreams, and it's also possible during hallucinations. So the question that really fascinates me is, when attention's withdrawn from peripheral vision, which do we think is more plausible? That there just aren't color experiences in the periphery, and we're somehow mistaken about the fact that it seems that we have color experiences in the periphery, or that color experiences continue, but they just might not be very accurate or veridical or a bit disconnected from reality at that point in terms of what's actually out there. So that's all the data that I have. Just a few follow-up experiments that we're planning to try and hone in on these questions. One is to obviously expand this work further into the periphery. We were limited by COVID conditions and needing to do online experiments, but we're hoping to do some in-lab versions of these experiments sometime soon. The second interesting question is that it would be nice to use more ethologically relevant natural images or natural image patches to see if the same sorts of dissimilarity reports are given with more naturalistic stimuli. And lastly, it would just be nice to be able to manipulate some attention and visibility conditions to see whether under different attentional conditions, participants give the same color similarity relationships that they do in this experiment where they've been constantly attending to the stimuli. And there's just some data from other students in our lab showing that the attentional conditions can make quite a difference to the similarity structures that we obtain from participants. So finally, I'd just like to thank everyone who helped me with these experiments and to thank all of you for your attention. And I'd be happy to take any questions.
Thank you. Thank you for the wonderful talk. So we take the questions from the audience. So please try to make your questions short so that we can take many questions. So first one from Shimojo-san, please.
Hello. Do you hear me?
Yeah, I hear you clearly.
Okay. So perhaps I should shut my mouth until we have time for discussion. But just for clarity, question, okay, and two brief questions.
Go for it.
Why only 10 degrees? Maybe your results had surprisingly disappointing similarity between fovea and periphery because it's only 10 degrees. So that's one question. The second question, why did you go with similarity? You might assume that similarity is inverse of discriminability, but similarity might be influenced by language, culture, various things. Inverse of discriminability is similar, but discriminability is much more, hard-on psychophysics taste. So, of course, it might be nonsense to ask for green and red discriminability, but you can degrade the presentation time or size or anything like that. And then you can try the same thing with discriminability. So those two questions.
Yeah, those are good questions. So the first question, why only 10 degrees? 10 degrees essentially because that's about as far as we could go while consistently being able to show the full stimulus patch at the peripheral magnified size that we needed on a participant's laptop from their standard seating condition. So about 50 centimeters away from the screen with a laptop screen that's maybe 30 to 40 centimeters. We can't push it any further than that without the stimuli starting to be cut off by the edge of the display. So it's just a practical constraint there. And that's why we're hoping to follow that up with in-laboratory testing where we can go much further out into the periphery. On your second point, why do we use similarity ratings? There's sort of a couple of ways in which I could answer that, but the main one would be there's sort of in a philosophical sense, it seems that similarity ratings capture something very essential about the subjective nature of experiences. So when you judge the similarity between different experiences, it seems like you're asking a subjective question about the experiences themselves. Whereas if you're asking about discriminability or detection, that seems less concerned with actual subjective experience and more with cognitive behavioral performance that you could imagine doing completely unconsciously. Obviously, it's not like there's not a cognitive component to similarity ratings, and it's not like discrimination can't reflect subjective experience. So it would be probably worthwhile doing the same sorts of experiments and instead asking for discrimination to see if there's actually anything special about similarity as opposed to discrimination or any other metric that you could imagine.
Okay, moderator.
Yeah. So any other questions or comments? Yeah. Okay, so Angus, please.
Yeah, hello. On that 10-degree point, this is a small thing, but you said that you picked 10 degrees based on standard seating positions from laptops. I was just wondering if there's any ethical concerns with getting participants to maybe sit closer to their screen, in which case you'd have more degrees of visual angle to play with.
So that would be nice. The issue with online experimentation to a certain degree is subject compliance over time. And it's very easy to imagine that if you're getting participants to sit in sort of weird ways compared to what they're used to doing, that they will start off doing the task properly, but slowly drift away. And there's all sorts of measures we can do to try and track subjects' attention, but it's a lot harder to track their seating position and viewing distance over time. We could do things like sort of tests for their blind spot again, or potentially even do eye tracking through webcams to try and make sure we have a sense of where they are. But it does get technically more difficult if you're asking them to alter how they normally sit.
I see. Thanks.
Any other questions or comments? Okay. Oh, yeah. If not, so we now move on to panel discussion with Dr. Shinsuke Shimojo from Caltech.
But there was one question from Nishida-san.
Okay. So, yeah. So please go.
After this, let's go to Shimura-san.
Nishida-san, please.
Thank you very much. So my question is about what kind of effects of difference between, what kind of difference between the central and peripheral you would expect. So since the situation is very simple here, so is there any prediction of the obviously the accuracy, precision may be reduced in the periphery at some stage, but probably and also the S-cone distribution and the other LM-cone distribution is different, so that there may be some difference in the hue in the, when you compare the central and the peripheral. But I think there are many potential things. But so can you tell me more about what kind of effect you would expect or you would like to see, and what?
Yeah, so I guess my point is that up until like very, very peripheral locations, I would expect the color structures to actually be the same, because it seems naively to me that I have colored experience all the way out into my peripheral vision. And we know that it's possible for parts of the brain to be pulling information from cones over quite large areas. And the point of this study is just to say, when people make the point that we don't have peripheral color experiences, or our peripheral color experiences are actually illusory or very different from our central color experiences, this is the saying, sort of in a sense, trying to look at the opposite and just say, actually, there's some evidence that our naive experience of colors being the same in the periphery, there's some evidence and experiments that we can design which would show that that actually is correct or potentially correct. So I really don't expect differences, except for maybe getting very far into the periphery where I'm not so sure anymore that my experiences are the same. And I'd have to look at the retinal neurophysiology to check that. I know that you get differential drop-offs in color-sensitive cones for different types of cones. So maybe we could make some predictions there about the exact effects that would happen.
So as for the perception of color, you need to consider the effects of calibration or adaptation to the world. So the after, for example, change of the color of a cornea, or the kind of aging effects can be calibrated over age. And probably even in the central and peripheral, even when there are some factors that may cause potential bias between the center difference between the center and the peripheral, that may be calibrated. But for example, if you can use the system like Professor Cohen to give a different kind of adaptation to the central and the peripheral area, and that may produce some bias. So in a natural environment, I think most of the factors will be canceled out by adaptation of the world. So I'm happy with your results, but if you want to see the difference, probably there's some kind of structure. It has to be introduced to potentially produce the difference between the central and peripheral.
Yeah, I guess I'm not expecting to see a difference. So what would, I guess, be helpful is if people think that there should be a difference in color experience in the periphery, to suggest what some of those differences might be, and then we can test for their existence.
Maybe I'll just add one more comment to that. As Ariel said, that we basically tested with this two naive hypothesis versus neurophysiology based kind of hypothesis, and then we found that our naive experience seems to be more or less supported by our data, but we also expected that there should be some kind of in a sense weird individuals, maybe as you said, that there is some kind of cataracts defect or peripheral cone deficit or something like that that could show up as an already similarity structures. And Ariel has already tested 73 people online, and we are extending this more people to do this. But there seems like some subjects seems to be, showing some, weird kind of structure, and we want to, get to nail of that. And we are asking some kind of questionnaire at the end, right? Even if it's rough,
yeah. So
blind people and so on.
That's right. You would expect that with large enough numbers, you would get some colorblind participants, and you would expect that the relational structures of these colorblind participants might be different to those with neurotypical vision. So we'd like to look at more populations like that with larger numbers.
Yeah.
Okay. Thank you very much.
Thank you. So now it's time to move on to the panel discussion session with Dr. Shinsuke Shimojo from Caltech and speakers and others. Yeah. So.
Okay. So do you hear me? You hear me, right?
Yes. Yes.
Okay. So I assume we have a little bit of time. So let me start with one thing, which is that I really want to explain why in the periphery, the mere physiological expectation is it's getting less and less saturated in color towards the extreme periphery, but we do phenomenologically don't have that feeling. And the first, Michael's talk directly addresses it, but the second Ariel's talk should address that too. So I want to have your opinion about why we don't feel in the extreme periphery it looks monochrome in the daily life. And I do want to, based on that, I do want to present alternative accounts to both of you. So that's my plan, but I would like to get feedback as to, in the periphery, phenomenologically, I'm looking around this room. The fovea, of course, is full color. And in the periphery, I don't feel like it's gradually monochrome. So why is that? Both of you.
Like us, Shin, you want us to just kind of tell our personal view?
No. So phenomenology is that, you agree, right? So there's but if you do discrimination, for instance, color discrimination experiment in the periphery, then, most likely you lose it unless you do cortically magnify. So you do have discrepancy between your subjective experience and actual psychophysical performance across colors. And I think your talk is very close to that phenomenon, and you might have an account. So,
I mean, I'm certainly happy to comment on it a little bit more. I think there's a couple different factors that are going on. I don't think there is one single response. So, for example, one reason that I think is because of the fact, this is going to sound a little out there, but bear with me for a split second. One reason I think is because people don't really necessarily often realize how many times in a second or a minute they are making saccades in the world. That saccades happen so quickly, so effortlessly, so easily. Stan DeHane's lab has even done some work showing that people underestimate how many saccades they make in a given period of time. That I think that intuitively a lot of people, I don't think this group, I think a lot of us, study vision, so probably are aware of this, but I think where that initial sense of folk psychology of a risk perceptual experience comes from is because you're like, I have an orange on my desk, and if it's over here in the periphery and I can't really see it, I can just look over real quick. And I think that action of me looking at the screen and then looking over in those two successive samples, I think people a lot of times don't realize that those are two successive samples that they are taking. And that they can easily overlook the fact that they are having to take multiple samples to have this knowledge base build up. And so I think that a lot of times people will confuse how easily information is available with it being something that they were perceiving the entire time. And then another reason is I think that people, and then I'll stop. And then another big reason, I think I kind of mentioned this before, is that I think people really oftentimes confuse knowing that something is a certain color in the periphery versus seeing that something is a certain color in the periphery. So I'll pull my orange out again. I've got this orange. It's over here. It is, when I put it about arm's length, right about there. I can't really see it anymore. But I know there's an orange over there. I can see something moving even, but I can't really make out the color. But people have I have this knowledge of this stable world where the world is in color. And I think a lot of times people are just like, I know what color the sky is. I know what color the grass is. I feel, and look, I'm right. When I look over there, I'm 100% right. And so I see all the colors that are out in the world. I don't even understand what the problem is. And that there's all these opportunities with the way that our visual system has evolved that we are able to take these really successive snapshots of perception, build them up into this knowledge base, and that gives us our impression of the world around us, even though I would argue, I know some people here would disagree with me, even though I would argue that our immediate impression or our immediate perceptual experience is less colorful than we think. We have these sort of heuristics and tools and tricks that allow us to kind of have the impression that we're seeing more than we have. This is just me speculating at a very high borderline philosophical point, but since Shin asked, this is how I kind of, at least right now, am biased towards thinking about how to reconcile intuition and this empirical data.
Yeah, so I like all you said, including your data, and I want to follow up and then, suggesting alternative accounts.
Before that, can you give also Ariel to respond to you as well?
Yes, so I just wanted to give a chance to him before I do that.
Yeah, so I, to a large degree, agree with Michael about how perception is different in different parts of the visual field. But I just want to make clear that there's not necessarily a one-to-one connection between perceptual information coming in the sense of external information that's available and the experiences that individuals have. So there's a difference between saying, when we say there's no, like you desaturate the periphery and you say that their perceptual experience is illusory or wrong in some sense, it could well be the case that they do have peripheral color experiences. They just no longer really correspond to what's out there. And it's the same point for if they have altered neurophysiology or they have any of these other issues in the periphery. That doesn't necessarily mean subjects actually have different experiences. And it's quite hard to capture what their actual experience is, but it's not necessarily the same as what you would expect based on just the information that's available to them.
Okay, so I would like to, propose some alternative accounts and see how you react. And then I probably shut my mouth to give a chance to others. But not against Bayesian, but highly overlapping with Bayesian, but perhaps different account could be, one is failure of change detection. And I think Michael mentioned this just briefly, but in a lot of cases, especially in the periphery, you just fail to detect the differences in color or anything else. And that might explain a lot of the data to me. Is this consistent, or is this entirely the same as Bayesian? I think it's mostly consistent, but I'm not sure entirely the same. The second account, which is related, is the brain compensation mechanism. And by this, what I mean is, like in the periphery, the input says it's black and white almost, but you would like to compensate to be a more consistent representation with where you attended to. And this is similar to size constancy, brightness constancy, all those things where our brain is trying to compensate its lack of accuracy in some sense. So those are my favorite account, and that's highly consistent with my observation in the periphery that, phenomenologically, I don't feel like it's desaturated, but, psychophysical performance is pretty bad. So do you agree or not? Is this entirely the same as Bayesian?
I would say, I feel like your account, the second thing you were just saying, Shin, was very consistent with, if I understood what Ariel was saying about this idea that perhaps, if I understand both of you, I think what you're basically saying is like what could be going on is that the input is not fully saturated, but your brain fills in these details, fills in this information. Okay, this might shock some people who know me that I'm about to say this. I would be so thrilled if that turned out to be the answer, because I would love it if you actually, I think for 25 years, 30 years, I don't know how many years, there's been this discussion about why is there so much data that suggests we see a significantly less than we intuitively feel like. And if the answer was because our brain is filling in these details and we're perceiving that filling in process, that input comes in, but it's that filling in perceptual process that we actually experience, I'd be like, oh, beautiful. That is the answer. This is how we can reconcile this data. And so now it opens this whole avenue of research. I have been thinking about this for a while, and I don't know how we test that. I don't know how we prove it. I don't know how we verify it. I don't even know where to begin with starting to think about what the experimental paradigms could be, because what does it mean? How do you verify that a person is not perceiving a stimulus as color? I have an orange. How do you guys verify that I'm not seeing the color orange from the external world through my eye, but I'm seeing it from internal representations in my brain? And I do think that this is something of a little bit of a Bayesian account at a neural level, Shin, because it's this idea of our brain is using knowledge and priors to fill in our perceptual system. And I think it's an extremely tantalizing hypothesis. And I'm really not being facetious, or I hope it doesn't seem like I'm being flippant or silly. I really would absolutely love it if this turned out to be the case. I think that would be a wonderful breakthrough. I'm a little reluctant because I just don't know what is the most compelling evidence that you guys can think of this sort of high level top-down filling in. Of course, there's things like neon color spreading. Of course, there's these lower level filling in sort of examples. But this idea that my brain can do it at the high level that I think is needed to fill in the gaps to understand phenomenology, I'm a little skeptical, just because I don't know what really good concrete evidence is of that existing in the world as we know it now. And I don't know how to study it going forward. So I love the idea. I'm just reticent to know how to accept it because I just don't really see the evidence for it or know how to get that evidence for it.
I have two very, very brief comments before I let Ariel talk. Very brief. One, there are a bunch of psychophysical studies of constancy, and it's objective measure, and that gives hint to the mechanisms. If we accept those seriously, for size constancy, distance constancy, brightness constancy, even the color domain, there's constancy. And, there are a whole bunch of studies of illusions and all those objective measures. To that extent, I think you could do it. That's one comment. Second comment, we did study extreme peripheral color perception, and confidence level is very high, period. Ariel, you might want to say something.
My point just on Michael's previous comment is that I think the primary evidence is from phenomenology itself. So the idea that people are so convinced they have these peripheral color experiences should suggest that it's worth looking for these in the brain in some sense and trying quite hard to find them. I agree that it's very difficult to imagine an experiment which you can use to show in a consistent way yes, somebody is having an orange color experience in the absence of an orange being presented to them. But that is sort of, I guess, the ultimate goal. And it's the same sort of question for how do you know people are having experiences when they have dreams? Or how do you know people are having experiences under any circumstance where there's a disconnect? In terms of Shin, your point about subjective inflation of confidence in the periphery, I don't really have a good explanation or connection between that and the phenomenon. Yeah, I'm sorry, I don't have a good point to make on that beyond noting it is an interesting phenomenon.
Well, thanks to be honest. I'm going to shut up, but the discussion reminded me of Edmund Land's famous demonstration that if you shut off one channel out of RGB, you still see upper as red based on the knowledge and memory, and that's kind of related, I think. But I'm going to stop here, okay?
Now, do you want to say something?
Yeah, I want to say something. And as I expected, this is great discussion and interesting, and it's also controversial. First, I want to disagree with Shin completely. That you started off with there is a discrepancy between the objective, performance and the phenomenology, right? But I'm not sure whether this is actually agreed upon. For example, also, including Michael's, demonstration of the orange at the periphery kind of things. This is all dependent on how you structure the task. And, the way that Michael is, structuring the task is developing this kind of expectation that, there will be nothing, and over the 9 trials and then 10th trial of the critical trials, you show something. And on the other hand, Ariel's experiment is basically trying to cut off all the expectation or top-down kind of, effect, and immediately ask people to report on what they saw, but also it allows them to pay attention to each thing, right? So there is both, advantage and disadvantage on different kind of experiment. But then coming back to asking about, Shin's initial, assumption about whether there is any phenomenological versus objective discrepancy, I don't think we have actually a good paradigm to suggest that, one is contradicting to the other. What I'm saying is that, if the objective sense of, peripheral color vision is, so bad that, the size of the object, like, orange at the extreme periphery is not visible in terms of its vivid orangeness, then I don't think, phenomenologically, we also see it as vivid orange. But somehow we are confusing this, kind of knowledge, as you said, Michael, that, we see orange here and then brought it here. So there is an expectation and knowledge that orange should be there versus seeing orangeness over there. That's just a confusion of the category of the things that we are trying to compare, right? But if we have
that's what I meant by failure of detection, failure of change. And, just come back to the constancy example. what size constancy stands for. Retinal image sizes are different between two objects, but in the reality, when you reach and tactile, sensation tells you that it's the same size. The brain, the visual system compensates for the retinal differences, which is just by observation condition. And the same applies in this case to the extreme periphery color. That's what I'm trying to say. So you made a mistake there. I'm not contrasting objective versus subjective. I'm contrasting about color signals from the retina versus what's expected in the phenomenology of the color.
Okay, if that's the case, then that's fine. But I think, if you start to talk about objective performance and performance as a sort of human subjects and subjective reports, it's not that clear that, these kind of things hugely, diverge. I think, what we don't see in the periphery, we can't also report. And we also, that aligns with objective performance as a, human psychophysical performance, but the retina still may respond differently. If that's the case, that's fine.
Okay.
Is there any other comment? If not, then I might say something. But Michael, Mark, you wanted to say something?
Yeah, well, I had a comment, Mark Changizi here, more about the sort of evolutionary differences that one might expect for where, for example, Michael, your experiments would continue as you found them versus maybe where they would fail. You might expect that. So in the case you have both surface colors as well as more spectral colors that are being conflated in those experiments. So you can certainly imagine that the surface colors as you move around the world are almost always invariant. They're not changing, and so it would be fairly easy for the brain to expect just to keep them whatever you think that they are, whether you last sampled them as. But if you were translating, rather than just staying still, where even most of the spectral colors, the nature of the reflected light due to the illuminance in the room, etc. If you're just rotating your head, most of those things are probably still fairly invariant. But if you're translating, as your subjects were actually moving around, I think, right, they're actually moving about. You could imagine that were you to have an experiment that just removed the surface colors or just removed the illuminance related, I can't remember what the right term is, that the spectral reflectance, not the reflectance of the surface, but the illuminance related ones, that one of them is more, that the surface ones are more able to be ignored or not notice that you're missing it. Whereas the other one, suddenly you're moving around and you're expecting, for example, it's a silver, some kind of gold chalice, which should be changing its speculars, in this particular way, and not just in a luminance related way, but also in a color kind of fashion. And it's not, and suddenly it becomes noticeable. So as and much more generally for all kinds of perceptual stimuli and modalities, whether the invariant, the ones that do remain constant as a function of translation, they're the ones that you won't notice when they're removed. But you start, let's say the size, the general shape of a surface compared to, let's say, T-junction or other kinds of junctions which tend to slide past each other, and you might expect that would be a kind of interesting question whether one of these is more susceptible to these effects than the other category. And that's the end of the question.
Yeah, I totally agree. I think the actual serious bottleneck, in all sincerity, is actually the technical side of making those stimuli. I was just trying to think about how I would do that in a virtual reality type of situation, in a dynamic video. And very quickly, I actually have a hard time kind of understanding or even intuiting what that would look like. But yeah, I think that there's a lot of different sort of properties that we are expecting to change versus there are things that we're not expecting to change that I think are both from the sort of expectations that Now was talking about from an experiment. But I also have to believe there's a lot that are coming from our entire life history and our evolutionary history as well. And I do think that there's a lot of possibility and a lot of potential fertile ground to explore how these different sorts of expectations will play a role in what people will and will not notice, for sure.
Sorry, just along the line, I have a concrete suggestion to Michael on your experiment. In your experiment, you have a gradual gradient of saturation from the fovea fixation point to the periphery, but you abruptly change from full color to black and white across the time frames. So I suggest, and this is what I mean by the failure of detection, by making it smooth across temporal frames, you can get even more extreme data, and you can make it to science and nature.
Wait, when you say smooth, what do you mean?
So at the beginning, you have full color, right? And then at one frame later, the fovea remains to be colorful, but periphery is going into gray.
No, that's not right. Sorry. It actually does happen over time.
How many frames?
So,
over minutes?
it's I think it's 3 to 5 seconds.
So by manipulating those parameters and getting Mark's suggestion as to examine what is easy to detect, what is not so easy to detect, is the paradigm that I suggest.
Sorry, I missed that last bit. Like, what do you mean by what is easy to tell? Oh, so basically trying to do the type of thing where you can sort of systematically identify what are the things that people will notice and what are the things that people won't notice when they change.
By the way, I have one account for flash lag effect in my mind here, because one account of flash lag effect is that the synchrony between flash versus the location in the smoothly moving motion. This is not something vision is designed for.
Right.
You feel like you can do it, but the vision is not good at that, and there's no problem not good at that. So that's what Mark is sort of vaguely thinking, and that's what I'm thinking. Yeah. Sorry, I keep talking.
Okay, so shall we take a break? Or
yeah, maybe.
Yeah. Or if there is any other further comment, or if nothing, then I might also say one thing, just briefly. I also think that the contrast between these two experiments are also something to think about. Why we get these kind of divergent results. And one thing I'm really interested is the power of the expectation. Actually, a series of the Michael's experiments and others in attentional blindness definitely manipulates expectation, right? And in our hands also, attention and expectation seems to be operating slightly different, cortical mechanism. Like expectation seems to be operating even at the, really early level of the, sensory gating. Like, if you don't, expect things to happen, you almost seems to stop processing it at the, early level. Like it's like late versus early kind of, attention selection debate in the, old days. But attention, if you tell people that there will be something to happen, meaning that, removing this expectation barrier, and upon that, if you ask people to attend or not, then that seems to penetrate through the brain, and then get modulation happens more, in a sense, deeper kind of areas. So I think it will be interesting in the future to dissociate between the neuronal mechanism and the psychological mechanism of expectation and attention, and how it actually changes our experience. That's kind of a comment. And I wanted to hear Michael's opinion about that.
So we wrestled with this issue so much now of like, what expectations should we give people? What instruction should we give people? What tasks should we give people? And we were really struggling. And this is actually what led us to just take a totally like agnostic view, where we were like, we're going to do none of that. We're going to not give anyone anything. We're going to put them in a situation and let them do what they want to do, because that to me is what is normally happening in everyday life. When you're standing there having a conversation with someone, when you're walking down the street, sometimes you do have a task and you like dig in on the details, but sometimes you take in like the vista and zoom out.
Yeah.
And I totally agree that those expectations and that attentional allocation is going to make a huge effect. That's why we did that later thing where we tell people, look for it and look for the desaturation. We noticed that the window got bigger. But this is just why we very intentionally didn't give people a task whatsoever, is because we were trying to actually capture the phenomenology of what's going on in everyday life. Because as soon as you start thinking about it and reflecting on it and attending to it and putting oranges in the periphery and so forth, to me, that is a little unnatural sort of state. That's not what our visual experience is normally like, because our attention is usually not to a single object in the periphery. It's normally, I don't know how to describe how it is normally allocated across the world.
Yeah, I agree with that. But, at the same time, we are now then coming into the realm of, the issue of what is a resting state or what is a normal state. And then we are also doing this kind of experiment where we directly ask people, what were you thinking? based on the probe that comes in a random, timing. It's called the experiential sampling. And in many cases, it turns out that, 20 to 40% of the time, people actually report that I was thinking about something else. Mm-hmm. And our recent paper shows that that kind of, report is corresponding to or preceded by the slow wave activity that happens in the, brain locally. Meaning, maybe that kind of thing happens. You actually do not maybe, process the things in the brain. So, we might need to directly ask people, even under the situation where there is no task, you kind of mentioned, right, that some subject actually spontaneously reported that, no, I was actually thinking about something else. And under that situation, maybe memory or report is totally, distorted, but actual experience itself might not be distorted.
Go ahead.
I was just also curious as to when you ask participants to attend to the periphery, Michael, I wonder how good a job they actually always do. And I'm curious as to what your thoughts on that are, because if you're in this virtual environment with all these like interesting stimuli, and particularly if VR is pretty new to you, it's very easy to imagine that you tell them to attend, but they actually end up like sort of mixing their attention between the periphery and then back to the center and the periphery and back to the center, and whether that makes whether you'd have different results in like some of your other paradigms where they're just stuck looking at a boring computer monitor and it's probably easier to attend to the periphery.
That is a fair point. I do think people are pretty good at it because, now I'm totally, this is going to be like anecdotes. Like people will sometimes be like, darn it, when I tell them that they missed the desaturation, or when we looked at their fixation patterns, the fixation patterns in the inattentional blindness version and in the attention to the periphery version were very, very different. And where people would look in a scene would be very different. People would look at the parts of the scene that were more color informative when they were trying to find whether or not the desaturation was occurring. So, but is it still possible that they're going to be like, hey, look, that's an interesting guy, or there's a rhinoceros or something, and that's a little distracting? Absolutely. That's definitely something that I can't totally negate. The one thing I just do want to make sure that I said in response to Now's point is I hope I didn't give the impression that I was trying to say that by virtue of not giving people a task, everyone was doing the same thing. I think Now is very correct to point out that you could have two people in the same exact scene who are given the same instructions in terms of just look around, and one person's really stressed. They have an exam tomorrow. They got in a fight with their spouse. They're in their own head. Whereas the other person is like, oh, wow, look, that's a rhinoceros. And so there's definitely a lack of control there. But for us, we just decided to be like, you know what, that's what's also going on in real life. So we're just going to kind of lean into that variance. But I definitely agree there is a lot of really important variance that's going to determine what you will and will not notice as a function of what you are, what kind of mental state you're in, for sure. There's no doubt about that in my mind.
Sorry, since now started comparing two experiments, I have a very, very short, concrete suggestion to the next experiment by Ariel. Okay, so just manipulate the difference only in color saturation. In one group, subjects are informed to pay attention. The other group, no, just natural passive and any difference. This will connect and compare the implication of the results with Michael's data.
Yeah, that's an interesting idea. So sort of duplicate the trials, but with some desaturation. So I'll keep that in mind. Thank you.
Okay, thank you for the stimulating discussions. And now it's time to close this session. And I'd like to thank the speakers again for their wonderful talks. Thank you. So
okay, now it's time to start second session. Second session is evolution of color Qualia. I'm a chair of this session. My name is Yusuke Moriguchi from Kyoto University. So first speaker is Professor Mutsumi Imai from Keio University. The title of talk is The Process of Color Lexical Acquisition, Its Implication for Symbol Grounding Problem. So Professor Imai, please start.
Hi, everyone. Thank you very much for inviting me to join this exciting symposium. So I'm talking about color lexicon acquisition today. the way different languages classify and label continuous color is extremely diverse. For example, these are two languages that do not have the distinct label for blue and green. So there are more languages in the world that do not have distinct labels for blue and green. Even though they have only 5 labels, the way that they divide the color, continuous color, is pretty different. And even among languages that seem to have very good correspondence in color categories, it is often the case that the same color could be labeled differently. For example, Dutch and German are very similar languages, and their color words have very close correspondence. But even here, there is difference. Dutch speakers label the center of the traffic light orange, whereas German speakers label it yellow. And a study showed that the language-specific labels affected the memory of the color, such that Dutch speakers choose more orangish color than what they actually saw when asked to choose a color that they had seen in this context, but not in a different context or context-free cases. So the structure of the color lexicon does seem to affect perception of color. This is a Whorfian effect, or relevant to Whorfian hypothesis. And then this is kind of a thorny issue. There are two parties arguing for the universality and language relativity on color perception. But, people think that the way their native language classifies the color continuum by words is the most natural and then only possible way of segmenting continuous color. So in this respect, acquisition of color lexicon is puzzling. On one hand, it's been demonstrated that infants show categorical perception much before they learn color words, or at least they start to produce their first color words. So for example, Yang et al. Showed that prelinguistic infants' brain responded to the color change from adult sense of blue to green, or actually it's in Japanese, so from Midori, between Midori and Ao. But on the other hand, color lexicon is diverse across different languages. In fact, as I noted, in fact, there are more languages that do not distinguish green and blue by label than those that do. So there are two views. There are two views for the account of color lexical acquisition. One view argues that what children do is to map word forms to already segmented color categories, right? So the acquisition should be pretty simple. But the other position argues that children have to find the particular way their language divides the color using the distribution of forms across context in the linguistic input. But neither position can fully account for the color lexical acquisition. The mapping view does not address how children eventually adjust themselves to acquire the language-specific lexical system. But on the other hand, the input-driven view does not really address how children can initiate the mapping process and then learn their first words, assuming that they cannot learn all color words at once from the beginning. In other words, satisfactory theory of color lexical acquisition needs to provide accounts on two respects. How is, in one respect is how is language specific lexicon created? That is, how is children's initial color lexicon like? And then how do children adjust their initial non-linguistic color categories to construct the language-specific lexicon possessed by adult native speakers? So to address both questions, we conducted a cross-linguistic study of color lexical acquisition targeting Japanese and German. Here, what we have to keep in mind is that the meaning of a word cannot stand on its own, nor can it be acquired independent of other words. For example, exact meaning of English word blue cannot be acquired without knowing how blue is different from green and purple. Likewise, exact meaning of owl cannot be acquired without knowing owl is distinguished from mizuiro, light blue, midori, green, murasaki, purple, and kon, navy. So, but it is obviously not possible for children to know all color words in the language from the beginning. So, so to study children's word, the process of word, color lexicon acquisition, we had to study adult lexicon, how adult lexicon are like, what the commonality and differences. We chose Japanese and German, which are not so dramatic than Berimmo or Himba that I showed. But here we wanted to see how do Japanese and Germans, their children learn the meaning of basic color words and then are immersed into the language-specific system of the color lexicon. And we are particularly interested in knowing whether the process, this process of color lexicon acquisition, same or different between the two languages. So, so we first, okay, so our experiments are very, very simple. So we tested German-speaking adults and Japanese-speaking adults, and we chose stimuli of 93 color patches from the system developed by Japan Color Research Institute. Okay, and the procedure is really embarrassingly simple. We presented each swatch in a random order and then just asked what color is this, eliciting their naming response. Okay, so here we identified basic colors for Japanese and German speakers. Well, so we considered here, instead of determining what basic color is, we considered the word as basic name for the language. If speakers use the word dominantly, at least for one swatch. So, so unlike other studies, in this study, non-monomorphemic words or words derived from source object, such as hellblau, light blue in German, or hadairo in Japanese, were included in the list of basic color words. The words marked in yellow were usually treated as non-basic words for English. Okay, and then we created similarity metrics for MDS. And from the name data, similarity matrices were created for Japanese and German. Okay, I skip the procedure because I'm sure that you all know. So 2 panels show MDS configurations for Japanese and in German. Each point represents each color swatch. The farther away the 2 points are, the more dissimilar they are. And we can see that relatively, there is relatively good correspondence at an individual word level, but the degree of boundary consistency among different words is different across Japanese and in German. So, so where are they different? Well, actually, what the MDS shows is that the size of the clusters on the MDS brain reflect the degree of individual inconsistency in color naming. In that the distance between 2 points on the MDS brain indicates the degree of disagreement in naming. For example, the cluster of cha, brown, on the MDS brain is larger than that of ao, blue. This suggests that there is less agreement in the category boundary of cha than that of blue among adult speakers of Japanese. And for each word cluster, we averaged the Euclidean distance between the centroid and each member of the cluster. And category size, here we made a graph showing the naming inconsistency and in category size. Category size is it's very simple. It is of a given word. Category size of a given word was calculated by counting the number of swatches that was labeled by that word most dominantly. And there is a correlation between naming inconsistency and category size, but as you see, it's not perfectly, the correspondence is not perfect. Okay, so here's the characteristic of in Japanese and in German. So we saw some common patterns, right? So there are the red is the narrowest and clearly separated from surrounding word boundaries. And then there is generally similar on the distribution of category size. And red and then yellow colors cover narrow ranges, and purple, blue, and green colors cover broader ranges. But here we see some differences between orange, the application of orange and brown. So orange is broad or broader in German and narrower in Japanese, and clearer boundary in Japanese, but the boundary is kind of inconsistent in German. On the other hand, brown is broader in Japanese than German, and inconsistent. The boundary is more inconsistent in Japanese than German. Also, German, we see some language-specific names, and in German has 2 names for pink, rosa, and then pink. And also, Japanese have 2 names for blue and mizuiro, mizuiro and ao. So we wanted to see how the difference in across the 2 languages in terms of the structure of the color lexicon might or might not affect the acquisition process. So we tested 3- and 5-year-olds, monolingual German-speaking or Japanese-speaking children. The stimuli are the same, and then procedure was the same as adults. We just simply showed each color swatch and then asked them to label. Okay, so here we see that, and actually, for to save time, instead of going through the details of the chart, I will just say the general trend in words. So white, black, red, yellow were well mapped to the typical referent in both languages. Gray were the color, the label gray, were learned late in both languages. And language-specific basic words such as mizuiro, kimidori in Japanese, or hellblau, turquoise in German, were learned late. But more interesting was green and blue were mapped accurately at 3 in German group, while Japanese children did not do so well. So in general, German children performed better than Japanese children. But one part of the color that they didn't do well was pink and rosa. So pink was mapped accurately by 3-year-olds in Japanese, but German children, 3-year-olds, did not name pink and then rosa accurately, both words. So here is the MDS configurations. So we conducted MDS slides from children's naming patterns, and as you can see, children applied the names much more inconsistently than adults did. It was not that there was no structure, but the boundaries are very largely overlapping. Here, German-speaking 3-year-olds named blue and green very distinctively, whereas when we look at Japanese, blue and green are almost, very largely overlapping. So, and in these panels show German speaker, the 5-year-olds pattern. Okay, so as you can see, 5 years configurations were in both languages much more similar to adults, right? So we can see a big developmental separation here. And in the precision or precise application of words. And then it is interesting that by 5, language-specific words like hell blue or mizuiro and kimidori emerged, or they started to produce them. 3-year-olds even didn't produce, okay? And the emergence of these words, language-specific words, pushed children's lexicon towards the structure of adult lexicon in language. We quantified how the accuracy of quantify the accuracy of boundary delineation for each word using as F-measure as the index. F-measure values represent the degree to which each child correctly assigned a given word term to the swatches without over or under extending word meanings. Okay, and here, again, instead of going through each colors in detail, we showed, we just summarized, the trend, okay? So what we found. So the colors like red, yellow, green, blue, brown, purple, pink, and then orange, those English sense of basic colors were learned earlier than language-specific words in terms of in terms of initial mapping. And language-specific color words converged on the adult usage slowly in both languages. So hell blue and then turquoise in German, and then mizuiro and then kimidori and hada in Japanese are actually they emerge late at the initial mapping, and then boundary delineation was through too. And so it seems like semantic structure affects the ease of learning. So to quantitatively test this, we conducted a model. We used two dependent variables. One is the accuracy in naming the typical instance of each word. So we tested the mapping words to the category centroid. And the second dependent measure was the degree of achievement of proper generalization of color word names. So with these two dependent variables, we had 5 independent variables. One is input frequency, naming inconsistency, category size, few uniqueness, and age. For few uniqueness, so we contrasted unique few colors like red, blue, green, yellow, and the others, non-unique few colors. What we found was, so here I show the result table, okay? For centroid mapping, what we found was age was important, and input frequency made the largest contribution, and then some category size in some degree. But boundary delineation, these factors, category size did contribute, but age didn't contribute, or input frequency didn't contribute. And what contributed was naming inconsistency. So this means, so naming inconsistency affects the acquisition of proper generalization of word meanings, but not affect the acquisition of typical reference. And surprisingly, perceptual salience did not contribute either for centroid mapping or boundary delineation. But it's probably because unique few words are said more frequently in the input. So it was probably eaten up by input frequency. So the summary is that we found 2 different mechanisms behind for centroid mapping and boundary delineation. The order of acquisition of individual words varies, but the underlying mechanism of acquisition of the color lexicon is common across languages, it seems. And what we found was across languages, children initially map core colors, white, black, red, yellow, blue, green, 2 words. But even for the initial mapping, language-specific difference in category boundaries affects the ease. And for boundary delineation, words adult speakers use with consistent boundaries are easier for children to learn, and words whose boundaries are inconsistently named are difficult to learn. So the conclusion is that children start learning of color words relying on biological categories, but even when mapping some basic focal colors to the corresponding word forms, children do struggle, and language-specific characteristics affect this initial mapping process. But children are extremely versatile and adaptive, being able to recruit useful properties in adult input, such as word frequency and then consistency with which each color word is applied, to assimilate the language-specific lexical system. Okay, so this is it. Thank you very much.
Simon, thank you so much for your interesting talk. So if you have any questions, Shimojo-sensei, please start question.
Yes. Okay, you hear me? Yes?
Yes.
Yes, we can hear you.
Okay, so this is sort of like an arranged question because Imai sensei sent me her earlier slides and I raised this. But this is definitely reminds us of the textbook level question as to whether language first or perception first. And then there are lots of studies in the past. So I asked her to give us a little bit broader introduction of the background. And I thought that would help the audience, not just me. So maybe can you just explain us?
Sure. So assume that the audience would know the term Wolffian hypothesis or linguistic relativity, right? Oh,
No.
Oh, okay. So I'm talking to a totally different parties, right? Different type of audience. Wow.
Well, your explanation made sense to some extent, even to me. But please go ahead.
Okay. So psycholinguists have studied extensively whether language influence our thought. And color perception is a very popular domain in this genre, right? So there is a lot of literature on the question of whether influence of language, that language affects color perception or memory. And in the classics, there is a very famous study by Eleanor Roche. Do you know or you don't know? Okay.
I know, but maybe just briefly, please.
Okay. Well, Roche studied Dani. I think it's a language spoken in a small village in Papua New Guinea. And here in this village, or speakers of this language, they have only 2 languages. So they actually do not have chromatic color words. And then they only distinguish light and dark color names. Okay. And so if you take a very strong position that language determines perception or thought, which in a way, a social linguist, Edward Wolff, no, I think it's Edward Sapir, and I forgot the first name of Wolff, but he claimed that language determines our thought or perception, okay? So if you take an extreme position on Wolff's thesis, then you would expect that Dani speakers can't distinguish and learn nonsense level for focal English sort of focal colors, or they can't memorize the difference between different hue of difference between red and orange, for example, red and orange, because they don't have labels, right? So Roche studied that, and she clearly rejected the Wolff's hypothesis at that level. However, the following study found that it was more nuanced. So Kean Kempton showed that speakers of English and Tarahumara, a language spoken in northern Mexico, with a triad of 3 chips of different shades of blue and green varied in brightness. And Tarahumara languages do not lexically distinguish these colors, like the colors labeled as green and blue in the sense of English. And Kean Kempton conducted the odd one out task and found that it was English speakers who distorted the perception. So the 2 chips when the 2 color chips were in the same lexical category, their distance was considered more similar. And when it crossed the category boundary, the English speakers judged that the 2 chips were more different than it was. And interestingly, Tarahumara speakers didn't show this category distortion by labels because they don't have it. Okay, so this clearly shows that the labels could affect the boundary of categories, color categories. And then there are bunch of following studies, and so there is a lot of evidence for the Wolffian effect. When you use very sensitive measures such as ERP response or as a latency response. And
due to the time constraint.
Yeah, can I ask 2 quick questions? Or if anybody else, that's fine.
Quick question, please.
Okay, so very brief, 2 questions.
Yeah, please. Yeah.
On your MDS results,
Yeah.
this is a little bit of a devil's advocate question, so forgive me, but it is very difficult to understand whether we should surprise that German and Japanese data are very similar, or we should surprise that it's very different. It really depends on the default expectation, and it's very unclear to me what would be the default expectation, the pattern of results based on one hypothesis versus another pattern of results based on the other hypothesis. And so, there are individual differences within the races, within the language groups, and there are differences between the cultures. So how should we evaluate?
Yeah, I think it's kind of hard with MDS to compare whether German has clearer configuration or clearer separation than Japanese. What the purpose that we conducted MDS is the difference, developmental difference, developmental change from 3 to 5 to adults, right? Then we can clearly see the developmental trend, right? So as you say, it's hard, but it's hard to compare German and Japanese lexical structure on this, right? But it's interesting that even in 3-year-olds, some parts are clearer, very heavily overlapping in German children, but green and blue are clearly separated in German, whereas which seem to reflect German adult lexicon. Whereas in Japanese, green and blue are really very heavily overlapping. And with age, it gets separated more, right? So at this level, we can say something, I think.
Okay, I got it. I'll postpone some other questions to the panel discussion. Yes. Thank you.
Okay, thanks so much for your question and comment. And we will receive another question after Mark's talk. So thank you so much for Imai Sensei. Great talk.
Thank you.
Okay, we will move to next talk. Next talk is by Mark Changizi. The title of his talk is Color of Emotional Expression. So Mark, please start.
Great. Let's see here. Just sharing.
Can you share?
Hopefully you can see me. I can't.
Yeah, I can hear. I can hear you, and I can see you.
Great.
Yeah, please start.
Let me get us started here. Let me just start with I'm going to get back to Russell's circumplex space at the end. This is a standard space people use to sometimes place emotional expressions, place different kinds of things in terms of how much, how pleasant or unpleasant, and how activated or deactivated. And it could be a useful space. And in terms of colors, colors are very emotional, and we'll talk about, some of the reasons they might be emotional in a moment. But if you had to ask, where do they go? Would you put red here and green? It leaves you fairly perplexed, like where should they go? You might say, yeah, some of these maybe seem more activated, some more pleasant, but it's not obvious that these should be straddling this space at all. And what we'll see later is that there's a sense in which these colors are everywhere. And to realize that, you have to understand that emotional expressions are at least 4-dimensional. And I'll walk you through that. And there's a sense in which they're just sort of distributed because once you go 4-dimensional, you realize that there's little copies of these things all over the place. So I'll get back to that at the end. So what I'm going to do quickly is I'm going to quickly talk about the old color vision work that I did a decade or more ago. And I'm going to tell you how that motivated an idea on emotional expressions. And I've been following that up really for a decade or more since then. And specifically, that older color vision is for seeing skin and emotions and health and that kind of stuff. It hinted towards there being 2 main axes to emotional expressions, namely my strength and your strength. And I'll get into that. And that led to this new theoretical foundation for emotional expressions, and that'll be coming out in my next book next year. And we'll look at where these two dimensions fit within this broader new framework that I can't fully tell you, but I won't have time to fully go through it. And we'll begin to uncover some of the emotional associations that colors have, and then get back to Russell's space and where all these colors are within it. All right, so let's, the old idea on color and skin, why did we evolve? Why did we primates evolve this extra dimension of color? Just as a reminder for those in the audience, your dog, your bunny rabbit, your horse doesn't have color in the way that we do. It has color vision, but it has yellow, blue as one dimension. It has grayscale as a second dimension. And so there's just two cones. You've got a short wavelength sensitive cone and what we call an ML cone with a sensitivity that's peaked around 550 nanometers or so. Whereas we primates, some of us primates, have that ML cone, the way we describe it, has split into two. And it's peculiar. And what's peculiar about it is that if you wanted to have a generic spectrometer in your eye, you'd want your three cones to be uniformly distributed over the spectrum. That's the way your cameras are. Your cameras have three filters and they're roughly uniformly spread. That's the way birds and other vertebrates that have four dimensions, in fact, instead of three, it's uniformly distributed. Whereas our new one is right next to this old one, and that's a really strange thing to do. Now, the standard theories for why, for 100 years, they thought, well, maybe we primates have color vision for finding fruit, and then more recently, sort of a similar idea, finding edible leaves. Within the vision community, they don't really give a darn about really why we evolved it. I think that if you ask people, they would say it's just about, distinguishing between different kinds of surfaces, surface colors, and various things like that, but, they don't really care. And then there's another, theory that it could just be some kind of genetic accident, and there's no particularly good reason for why we have it at all. And it occurred to me more than a decade ago that maybe it has something to do with this. The kinds of colors that we, and even SpongeBob here, cartoonists add to their cartoons because it's so important for the way we think about other social creatures. And primates are well known, and we are, to show these kinds of signals, and they're deeply important signals that tells us a lot about the state of another. So to understand the kinds of signals that are coming off of our skin, you have to understand that the skin is actually transparent. You don't usually notice how transparent your skin is, but if you think about it, when you get punched, something happens, you get punched or you get a wound, not a wound, but just a bruise. Suddenly you see this massive bruise. So what you're seeing is the blood underneath the skin, which shows how transparent your skin is. So all the time when you're seeing skin, you kind of imagine that it's opaque, but it's not opaque. It's not opaque in the way that a mannequin at a store is. It really is that you're seeing through it to the blood below it, and it's constantly shimmering and changing its color and nature depending on where you're looking at the time that you're looking. So it actually undergoes two different kinds of changes that really summarize most of the kinds of changes, not counting bruising and these other kinds of things like this. More blood, higher concentration blood, this should be pointing to this blue here, and less concentration, because bluer when it's higher concentration and yellower when there's less blood in it. And then there's how oxygenated the blood is. When it's deoxygenated, this little W feature goes away in something. It goes, so when it's more oxygenated, it has this W feature in the skin. So what you're really seeing, these wiggles here, this is a skin spectrum, but what you're really seeing that matters here is how the skin changes due to the changes of the hemoglobin underneath the skin. So if you are a dog or a bunny rabbit or your horse or a typical mammal, you're not going to be able to distinguish between these two different dimensions of blood change because they're all, they're all redundantly, they're all along this thing. You can't distinguish it. What you want is to be able to have some kind of sensitivity here at the trough of this oxygenated W and at the peak. Therefore, thereby you could see whether it's oxygenated or deoxygenated and still have your old ML sensitivity and still be able to see the rise and fall of the concentration. And so when you think that through, that's what I was thinking 15 years ago. And then I said, well, this is exactly where the ML cones are. It's exactly where you need to be to have this peculiar sensitivity that it's the peculiar position you need to be in to have the sensitivity for oxygenation. So the idea behind that theory, I'm not going to get into more about that theory, but it led to this idea that redder and greener. The meanings of red and green really come by virtue of the skin, and that one is high oxygenation and one is low oxygenation. And really, high oxygenation is kind of an honest signal of I am strong. You can't fake oxygenation. If I'm in a fight with you and we're fighting and I'm ready to pass out, I can't fake having red skin. And deoxygenated is weak. So this I am strong and I am weak, or I am confident and I am not confident, this kind of axis seems to be quite important. But remember, it's not just one dimension. Skin not only changes along red-green, it also changes along yellow-blue. In fact, by virtue of this, skin, by virtue of oxygenation and concentration changes, can achieve any hue at all. And it's not because the skin is really doing it, it's because your eyes have evolved in such a manner that it happens to be that it's designed so that the natural states that skin can undergo by virtue of the blood achieves any hue. So once you realize that there's both of these dimensions, well, maybe the question that you might ask is, well, now that we started signaling with our skin, some tens of millions of years ago, and starting to show bare skin so that we can purposely signal these things rather than them being sort of just reading the side effects of what's going on underneath somebody else. Well, then you might wonder, well, what could it be? Could it be that we're signaling this as well, signaling yellow sometimes and signaling blue, that they themselves are potentially signals? And low blood happens, for example, when you're afraid. I have to do fight or flight. I've got to run. I can't afford to have any blood in my extremities. I have to push them down deep, into the muscles and into the stuff because I'm actually doing something. So you start to wonder, well, maybe this has something to do with afraid, and so then maybe this is like anti-afraid, something like disdain, where I'm not even a little concerned about you. So, and that's the idea behind that is, well, maybe this is about you are strong and you are weak. Now, this is all questions. These are sort of speculations in my mind, but I had this idea: could it be that this sort of the red-green is I'm strong and I'm weak, and maybe this other dimension, in terms of the meaning for what they truly mean for the associations of the colors, comes by virtue of the meaning on the skin. And in these cases, it's really about you. These signals are about what I deem you to be, your strength, and what I deem my strength is by virtue of that. So that got me thinking, this kind of idea. And let me just quickly reorient it to the way that I'll be showing it for the rest of the talk. So the idea was of these two key dimensions, just I'm strong and I'm weak, a my strength axis, and a your strength axis. And there was something just quite beautiful about that, just even outside of the color domain. It's just me and you and our strengths. And in social, could it be that such a simple, elegant axis could be the key dimensions for our emotional expressions generally? And that question would drive me for a decade, because that's what you want in social relationships. It's me and you, and we have some kind of disagreement. I'm confident to some extent, and you're confident, or have some strength to some extent, knowing how we would settle this if we couldn't come to an agreement, just like in poker, how strong our hands are. So it seems to have all the machinery you want. It's just two people and their strengths. And in fact, when you start imagining what emotions might go with it, 4 of the basic emotions from Ekman sort of pop up immediately. Well, if I had to put something with my strong amongst the 6 basic emotions, angry sort of fits here, and sad, if you had to pick one, the being weak would be here, and then fear we already talked about, and something like disgust. But more generally, the thought was this is more something like confident or proud, and something like humility or humble, and this is something more like respect, and this is more like something like disdain as a less, if I wasn't having to pick emotions that are amongst Ekman's, the basic emotions. So that's the question. So over 10 years, in some sense, this kind of idea drove me forward to looking for how can we understand emotional expressions, and it felt like this has to be the fundamental space that, in some sense, undergirds it. And I've just put in, eyebrows, sort of proud eyebrows here, and sort of weak eyebrows, and then a downturned respectful nose and an upturned sort of disdainful nose here, just to put a face to it. Now, these dimensions, I happen to have shown these things diagonally, but you clearly then get 2 rectilinear dimensions. When you think about this, you have, since this I'm strong and you're strong upwards, we have a force or a strength going upwards here. So the y-axis is like force or tension, and it goes from like I'm serious to I'm casual, as you go I'm serious down to I'm casual. And then the x-axis, you might call the compromise axis. it's like whatever the two people are, whatever it is that they're negotiating or arguing about, how good something is, or how much potato salad, I should have compared to you, whatever it might be. There's something that they're pushing about, and this is how much force there is. Now, this space, force, in this sort of this horizontal axis is actually a very familiar space. It's quite a simple one and a quite important one. So any space like this, you can think of as just two springs as one example within the space, and there's other reasons for thinking springs are important, which I'm not going to get into here. In springs, if I've just got B here and pushing on some mass, and C coming off of a wall and pushing here, C wants to push all the way to here. C wants to get this mass all the way over here, and B wants to push it here, but they're both stuck by virtue of each pushing at each other, and they come to some kind of equilibrium by virtue of it here. And if I was to have tightened this one and make it a stronger spring, then the actual force here would be higher. The position on this graph doesn't mean anything. It's just that the position would be higher. So, and I'm showing their spring constants here are really spring curves. These force curves are actually very similar to how humans push back. The more that you ask of me in a negotiation, the more you say you should, if I'm selling my house, or if I'm the buyer, the more that you ask for more and more money, the more I'm going to push back. If I'm the buyer, I'm this guy. I'm pushing for lower prices. So our desires in a negotiation are pushing against one another, and they're very similar to the way that springs are, because we push more and we're more dissatisfied the more that it goes in our disfavor. And in fact, so just as an example, Mark and Tim, I have a co-author, Tim, in this next book. Concerning the fraction of this zucchini loaf that Tim should get, I might believe he should get 1/3 of it, and he believes he should get 2/3 of it. And let's suppose that Mom, let's suppose we're brothers. Mom knows how much we're supposed to get, and we could always settle this debate by going and asking Mom, or wrestling about it, or something like this. But right now we're just having it, like trying to signal and express to each other, so we don't actually have to fight about it and go ask Mom. And the nature of any kind of negotiation like this is that I believe this, and he believes this, and he's more confident in this case. He has a little bit more certainty than I am of what Mom's going to say. But instead of thinking about it as just curves, you can think about them as these disbelief curves. So these are really 1 over the cumulative probability distribution. It just says that Mark is pushing to the left. I want less zucchini bread for him, and the more zucchini bread he gets, the more my force pushes leftward, just like a spring, and the same for him. He's pushing the other direction. These are just the springification of our belief curves, of our Gaussian curves there. And again, so it's just like my desires, or my will, is like a spring, and his will is like a spring. So that's the importance of these kinds of curves, is that these curves are within this force compromise space, the same space we got to before. And in fact, so the basic idea for part of an emotional expression, the part that I call a response, is just an expression of my claim about my confidence and your confidence. So my confidence, confidence, say yes. Did somebody talk? Okay. So my confidence, I might say, look, Tim, I'm this confident, and I think this is me saying my strength. I'm this confident, this curve here. This is my spring pushing to the left. And I'm saying, Tim, I think you're only this confident, whereas Tim was saying that Tim was, much steeper confidence, and he was saying I wasn't very confident. So I can say each of these, my strength and your strength, in terms of these sort of red, green, and yellow, blues, and actually put a point within this space. And it also, and then he could respond and I could respond back and forth, and we could eventually decide. But notice that whenever I say that, no, actually I'm this strong and you're only this strong, I'm also making a counteroffer. I'm saying, like, no, you only get this much and I'm this much serious about it. I'm having this much tension. And this is what happens every time, and people don't really realize this. If you're trying to buy a house from somebody, you tell your realtor to offer $105,000, but you also tell your realtor this other information. You say, and tell them this is my final offer. Or you might say instead, okay, $105,000, but tell them I am flexible. I'm still flexible, right? That would be giving them a low tension. Both of those things are communicated whenever we negotiate, even today, and they're in fact part and parcel of what has to happen in any negotiation between any kind of animal. So you can think about it as in this space as my spring constant and your spring constant, or how strong we are, or about here's my offer and here's how serious I am about it. And so that leads to these two dimensions, the same ones we started, still in the same space, the space that it was motivated by red, green, and yellow, blue, but as the general space that emotional expressions have to have. And then more generally, the idea is that, you know, if you're thinking in these terms, if I just want to say I should get more and I'm not changing it up or down, well, then that would be like purple in this case, but we're looking just at this, I'm using these as just these colors as metaphors now within the space. I would be basically saying, look, I'm more confident and I'm a lot more confident than you realize, and you're a lot less confident. Or I could just say, I should get more. So this space is in fact crucial for understanding emotional expressions. It's 2 of the 4 dimensions. Now I'm going to briefly tell you about the second pair of dimensions, and it too is going to be within this space. Now, and then I'm going to quickly show you the full space and then we'll be done. But in real life, between social animals, if I express to you and I say, look, I want more, then your first response, you need to acknowledge that I said that. Or let's say it the other way. You tell me, Mark, I want more, or you do this with an emotional expression. My first response is like, okay, let me get this straight. You want more? No way. Or you want more? Okay, you can have it. So I first acknowledge, I have to find some way to acknowledge or a confirmation receipt that I got the message, so that then that's how you maintain eye contact, you maintain the tension, so that you can be sure that I got the message so that I can go back. So suppose that you, in fact, were aggressive towards me, saying, look, I want more, and saying that basically you're stronger and I'm less strong than we realize. Well, I'd be unhappy, right? And in fact, I could signal unhappy to you. And my claim, the claim in this series is just unhappy is not me telling you how I feel inside. It's me acknowledging a receipt that I got your message that you want more. And if instead you were conciliatory, you said, hey, Mark, you can have more. Of course, inside I might be happy, but the reason I signal it is it's an acknowledgement that I got your message that you're offering more. And similarly for serious, if you just say, Mark, look, I'm not joking around, and I didn't expect that, I might say I show surprise as an acknowledgement that you said that. And same, if you say, look, I'm really casual, then I show that, oh, I'm just relaxed. I say, cool. I get a beer and I say, we're just relaxed. This is not a serious discussion. And again, you can end up with sort of terms in each of these corners. So in this way, you end up with 2 spaces. You end up with these acknowledgements, and you also end up with these responses. And the key thing is that both of these spaces are the same space that I talked about at the beginning. They're both the same force and sort of this compromise space. This one is just acknowledging what you just signaled in that space. That is, I'm just acknowledging what you signaled in that same space. And here I'm telling you what I think in that space. But it's the same space. And the key here is that an entire expression, the theory says the entire emotional expression is that, suppose that you just asked for more for yourself, then I would say, okay, you just suggested a worse deal for me. So I show unhappy as an acknowledgement of that. But then suppose I reply and say, look, but I'm much more confident than you realize, and which means I want more and I'm serious about it, then I might do this. My entire expression will be a combination of being unhappy as an acknowledgement to your aggression, and in this case, showing pride. Each combination of this plus one of these, always one of these and one of these, is what it is to engage in a negotiation between one social animal and another. It's always both of these. It's saying, you just said this, well, I say this, each time. So that means that there's not just these, there's not just 9, including neutral in the middle, and these 9, but it's 9 times 9. So how can we show all of these expressions? All of the emotional expressions that you get out of this are as follows. So one way to show it is that I'm going to show as the trunks. These trunks here in this space are the acknowledgements. So now we have unhappy and happy here, surprised and relaxed here. And then for each of those, depending on how this is basically saying what you just did, saying if I say I'm unhappy that you were just aggressive, then given that, here's how I respond, and so then I can move to any of these positions. So in this last case, it would be up here in this space because I was unhappy and then I showed pride, right? So you can end up in any of these possible combinations. I can, on the basis of the rules that I've been using here for the face, the brows and the nose, and in this case for surprise, it was sort of eyes open and mouth open to eyes shut and mouth shut. and here was just a frown versus happy. All of the spots here, by those same rules, now have a face. And if you go through one by one through each of these different cases, you can come up with approximate names that seem to describe it. And one of the interesting things here is that the one we just talked about, I'm unhappy and I showed pride. Well, in the case of being prideful and unhappy, it sounds a lot like angry. But if I'm unhappy and I'm showing humility, that sounds a lot more like sad. Whereas if I'm unhappy and I'm showing respect, well, now it sounds not like respect so much, but fear. And if I'm unhappy and I'm showing disdain, it's no longer just disdain, it's something more like disgust. And in fact, let's just look at angry. If I said I was happy, but showed pride, well, if I'm happy, you were just conciliatory. You actually gave me more. But I still say, I acknowledge that you're offering me more, but I'm even stronger than that. That's smug, right? That's very different. Or if instead you offer me more, but I say, and you're even worse than that, like that's sneering, right? So the notion of what it is to show disdain or what it is to show pride changes drastically depending upon the context of whether I'm unhappy or happy, which is the context of whatever you just did in terms of having been aggressively asking for more or less. Now let's go back to where we started, which was Russell. This is the same space that people often place emotional expressions or emotions and various things within them. And we can look at our space in the same way. Across the acknowledgements, we have unhappy to happy. So that is, when you go within these tree bunches, as you move across, you're getting to be a pleasant valence, high and low valence over here. And over here is more activated. Surprise, you're more activated. The person is very serious. You better things could go to a fight, so to speak. It might leave the discussion. Someone calls and poker, so to speak, and then you have to go figure out what really would happen if you weren't able to talk about it, versus being very relaxed over here. So we see this is how the actual emotional expressions fill this space. But let's look at how usually when people, if you're just interested in the 6 basic emotions, often this is where they typically roughly end up when you're doing this, putting them in this space. And then here is where they end up in our space, because surprise and happy were in those spots. Angry and fear, have this sort of same level of unpleasantness because they were both in the same box. And these ones are in the same box. These are a little higher because I won't get into the details here, but roughly you end up with the same spot as they end up in these measurements. But what it really shows you is how useless somehow thinking in those 6 basic emotional expressions are. Russell space is useful at capturing some of the things, but 4 of the 6 basic emotional expressions end up all on one side. However you do it, it's sort of missing the point. And then the dimensionality isn't 6 at all. If you're thinking about 6 basic dimensions, the problem is that you're likely to think of them as 6 independent dimensions along which any combination is possible, not necessarily equally possible, equally probable. But the dimensionality isn't 6 at all. It's because angry and sad are opponents within this unhappy case, and fear and disgust are opponents. And in fact, these 4 aren't just over here on the right. The key feature of these 4 is the fact that these are cases of pride and humility and respect and disdain, and they're spread all over the place. So the real primes in this space are just 4 primes, not 6. There's only 4 basic ones, or primes in this sense. We just have pride and humble, respect and disdain, which is exactly the same ones we talked about at the very beginning. I'm strong, I'm weak. You're strong, you're weak. And then we have these 4 primes, which is unhappy, which is basically you just said you want more, which can be fleshed out as you're strong and I'm weak if we go diagonally. And so we have unhappy, happy, and we have surprised and relaxed. Those are 4 ways of picking primes, which give you the 4 dimensions you need to get the entire space of these motions. But you see that the colors aren't, if you were trying to place where does red go, well, red is everywhere. Any spot within this space of Russell's, if you put your pin at any different spot in the space, then in that pin there's going to be some micro little universe of these response emotional expressions within there, anywhere that you go. And that changes entirely how you think about the emotional expressions associated with colors. It's not as simple as that. It depends, of course, on more. But what the key thing that they have is this I'm strong, I'm weak. you're strong, you're weak, and that's what's retained amongst them. To summarize then, so my research on the origins of color vision helped reveal what colors ecologically mean, at least for red-green. It was primarily about red-green. It was about my strength. I'm strong, red skin that's oxygenated, and to I'm weak, green skin, deoxygenated. And that made one ask, or that made me ask, what might the other axis of skin color change mean? It seemed to maybe be having to be something about your strength. You're strong, yellow skin, low concentration. You're weak if I'm blue skin and high concentration. And that results in two-dimensional space is quite natural. It's exactly what happens when there's two springs in a duel. And it's also what happens when there's two people in a duel, like, with differing opinions. And not actually when they're actually dueling with swords, but the idea is when their wills are dueling and then they're in a conversation trying to settle it without having to fight. And generally, negotiating a compromise requires that each participant signal in exactly those two dimensions. The x-axis is aggressive to conciliatory, and that's just how much I want. And the y-axis, which is sort of orange to blue-green, is how serious I am about it. And if you add these confirmation receipts, these acknowledgments that I talked about, you get emotional expressions similar. I mean, those emotional expressions in that big space that we looked at are plausibly very close to the kind that we have, retaining the kind of position within the circumplex space of angry, sad, and all the 6 basic emotional expressions, but having a much wider reach in terms of its ability to place all of these other emotional expressions as well. All right, thank you very much. That's all.
Okay, Mark, thanks so much for your stimulating, exciting talk. So given the time, we will directly go to panel discussion, and we will take Shimojo Sensei's comment, then we will receive audience's questions. So first, Shimojo Sensei, please start your comment.
Okay, so Mark, thanks for your talk. We have to save time, so 2 quick questions. The first one is trying to connect your talk to Imai Sensei's developmental talk, and I want to ask, these color emotional connotations are something to learn in childhood, you think? And is it structured by language? Or this is something you assume, since human type trichromatic color channels are genetically almost determined, do you think this is sort of like already there? So that's one question. And the second question is, even though you didn't talk much about Qualia, I wonder if you want to say anything about, your analysis say anything about Qualia scientifically. And by Qualia, I guess we are mainly meaning subjective as opposed to objective experience, and also explicit, conscious, as opposed to implicit experiences. So those 2 questions.
Yeah. On the latter, I certainly don't think of myself as a Qualia researcher per se. I don't think I've used that word probably in any paper or book. But I guess I would, from my point of view, if you want to understand the nature of the experiences of Qualia like color, then the way that I always imagine is that you really have to understand what these things mean, and understand what they mean, you have to go evolutionarily to know what's driving the evolution of these, having these perceptions in the first place. So for color, and that's true for both in terms of color, the question is what they mean, and the claim here is that it has something that has to do with skin and oxygenation, deoxygenation, and really the meaning there is about strength and things. That's really the key, not about oxygenation per se and things like this. And if you also want to understand Qualia, then you want to understand all the conceptual associations they have to other parts of your Qualia network, because those are what often make the Qualia feel the way they do in the first place. Red feels strong. Red is much more likely to be associated with a sword than it is a shield, for example. Why is that? It's because there's something aggressive about it. And if you want to understand the feeling and nature of these Qualia, you have to understand these fundamental meanings. And if you want to, so the claim is that by understanding color, how they associate with emotional expressions more generally, and there's other things if I can share for just a second here. One more second, I left to put a couple other slides here. Oh, I got to go through the whole good more all again. Let's see. So for example, the same space that we talked about in terms of, you can think about, you have to do preparatory states and emotional expressions. If I'm saying that I'm aggressive, now I'm only having a conversation with you that I'd like more. But if it turns out that we have to, if it turns out that that we decide to fight about it instead, and that can be asking mom or any number of different, doesn't actually mean we're wrestling about it, but I have to be prepared because at any moment I've just said that I deserve this much more. So I'm going to have to be willing to fight for it and say, Mom, I really, this really was, whatever it is the fight is, I have to be using my sword to go potentially get it. Whereas if I said that, no, you can have more, then whatever our fight is going to be about, I'm not wielding it against you. I'm kind of pointing it away. So you can actually sort of show these kinds of, in the case of, if you were aggressive towards me, then if we were having to fight about it, if me being unhappy, my way of it, by acknowledging unhappy, I'm kind of realizing like, oh, it looks like I'm going to have to put up a defense if we end up having to fight about it. So the metaphor association with that would be something defensive, and it's going to have to be raised or lowered depending upon these different states. So you can actually then work out, for each of these different states, they all have an associated sort of a warrior situation in terms of what their shield would be doing and in terms of what their sword would be doing, or viewed from a first-person perspective like this. So this kind of looks angry. Even if you took his face away, he's ready to swing it at you and he's defensive. Whereas here, he's sort of swinging it because he's smug, but he's not worried about you even swinging at him, so he has a shield behind him. All of these sorts of associations, these that happen, these are suites of associations that are connected to colors and are all intermingled with one another. And the only way to understand that is to sort of flesh out these sorts of grand unifying theories that tie them to the kinds of evolutionary needs in the math, in some sense, of what has to happen in a negotiation with two social animals and the kinds of signals and the kind of constraints and demands that they were under. That was
we should probably ask. Yeah, we should probably ask.
Yeah. Well, the first question was, I don't think language has any. I think certainly language is second to third order, and that these things are governing, primates generally, and all social animals. Of course, not for color vision, but all social animals are going to have these same emotional expressions, albeit they're not going to have the spectral ones.
Maybe we should also ask Imai-san to respond as well. You are unmuted. You're muted. Okay.
Okay, well, thank you very much. I don't know if I agree with the last comment. Well, children, if you are right, then it should be easy for children to learn color words and emotion words, right? And it's known that in both domains, these are the color words and emotion words are something that they struggle. They have really great difficulty. And then it's because, so language have different systems, lexical systems. And then children have to adjust that. And so as I presented in my presentation, even basic colors like red, blue, green, yellow, it's not easy for them, right? So if that is the case, I don't know if the mapping goes so naturally. And then I think it's an empirical question whether there is much universality across. It's a lot of anthropological work is required to support your claim, I think.
So on the association, the emotional associations of colors, right? On that portion of it?
Well, no, both like colors and emotion. Oh yeah, emotional association. So it's like color and then emotion are, on its own kind of very relative across languages. And also, what you say, the association between color and an emotion is one levels up. So it's even more complex and complicated. So I don't think it's...
There's a solution.
I certainly think that asking humans these things it will have throw in a lot of complexities involved. But the meanings of emotion of the colors on the skin of other primates will of course have the same ecological meaning as it does for us, and of course those meanings can be overridden in human culture, which is radically changes us in many different ways. I think that one is going to it's going to get noise, whole lot of noise on it by virtue of all the culture that we have piled up, which I'm appreciative, and I have books about that as well. But I still think at its core, the actual unadulterated by culture associations that these colors and emotions are about is about these sorts of strength-oriented kinds of meanings.
I have a compromise between you guys' two views. Mark is talking about implicit sort of intrinsic mapping. And Imai Sensei is talking about how to bring it through the network, a discrimination network of linguistic category to appropriately use the word in the native language. Those two are entirely different things to me.
Yeah, I think so.
Okay, so can we go to the next question? Okay, so please, Ariel, start your question.
Thanks for your talk, Mark. It seems like your theory makes some pretty strong empirical predictions about what skin color individuals should have under different emotional conditions. I'm wondering if you've done any experiments or know of anyone who's done any experiments to check whether what your theory predicts actually matches up with what people's skin color is when they're in particular emotional states. And sort of secondary to that, what makes me a little bit skeptical is that presumably a lot of human evolution happened when we had very dark skin or when we still had a lot more hair. So I'm just wondering if you could comment on that as well.
On the latter, the primates that have color vision, trichromacy, all have naked spots, naked faces, and other naked parts on their body as well. And dark skin, these things are all visible on all the different skin colors. They're all actually visible. Now, someone who's, let's say, Caucasian and doesn't have much experience with faraway ethnicities, when Darwin first was corresponding to folks that were on the other side of the earth, half of them would say, yeah, he was asking, do they blush? Do they do all these, things like this? And some of the stories that came back were that, no, they didn't originally blush, but after experience with us for 2 years or whatever like this, they began to blush as if they had become civilized and learned how to blush. But it's much more plausible that, of course, Caucasians not being used to seeing that skin tone were just not good at seeing. It was very far from their adaptation, so they just couldn't see the colors to begin with. On the first question, this larger theoretical framework that I'm talking about here, no one has, it's not even out yet. But certainly since the paper, the earlier discovery that I talked about at the beginning of the talk, there's been a lot of folks that have been looking at how changing the color in subtle ways in images, how it's perceived by viewers as having a particular emotion, which is consistent with that. Now, of course, So there's now a lot of evidence, and I haven't really been keeping track of those. And there's been a lot of interest in changing the colors of the skin and seeing how people respond. Yeah.
I just want to follow up and answer that question, because if you do psychophysics of skin color, subtle skin color, there are some evidence. So I think partially empirical or psychophysical evidence to support a part of Mark's argument. Some others are sort of looking seemingly inconsistent, but it's too complicated, lots of factors. But if you look at, I think Mark's contribution is that if you look at the cultural icons, and what color is used for what kind of smiley faces and stuff like that, and it's kind of obvious that what he said, the emotional connotation of the color is there, and it's not entirely, but pretty much cross-cultural.
Yeah, I guess my point's just relative to a neutral, not particularly emotional baseline skin color. It seems like this would make strong predictions that somebody would experience emotions and then their color would shift, of their skin would shift in a particular way, to really back up the theory if that experiment was done. So it'd be cool to see. Anyway, thanks.
Okay,
can I ask a question or maybe comments? I'm quite happy and also excited to see both Mark and also Imai-san talked about structure or relational structure of in meaning and also color and emotion, because as I mentioned in the first part of my opening talk, that this Qualia structure team is trying to understand the Qualia through characterization of the structure in relation to others, not only within color. This color symposium is just starting point, because we thought that color is probably most understood or studied through relational kind of context, but of course emotion and also how this structure is influenced by development and also evolution, and are definitely interesting kind of thing to think. And also naturally, in the end, I believe that all these relation structure or qualia is coming from the particular parts of the brain that has particular structural property that generates this specific color experience or specific emotional experience or linguistic kind of meaning. So this is all nice and also sort of kind of a interesting kind of a convergence, but also we realize that many different kind of fields have different barriers in concepts and so on. So we are excited to make further interdisciplinary conversation on that. So that's the kind of the comment on that. And then in relation to the sort of further sort of the exploration about this kind of thing, my colleague in Monash recently discovered that the meaning of the red seems to be preserved also for the monkeys. Like when he did a bunch of different kind of tasks on the monkeys who are trichromat, it seems like red is easy for them to associate with the sort of the stopping signal or inconsistency and so on. So there seems like red is particularly salient or meaningful, maybe even evolutionary trajectory. So I wonder whether you have any kind of conjecture about the origin of the color qualia potentially in the evolution or maybe development when we start to see this thing, experience these things.
Well, I mean, for me, I mean, of course, when exactly it happened, I would imagine to get a little bit more fine scale, probably there was some accident, some first primate had this special trichromacy and we hadn't co-evolved to lose our fur very much, but they could still use that to read, had a little bit of an extra empath sense and read, around the nose and parts where they could see. But over time, eventually it co-evolved so that there was a loss of fur, so that then the animals are starting to signal on purpose. And so the meaning during that time, somewhere along that time, the what it's like to see red would have come to have a particular ecological meaning, namely the person that I'm talking to is showing a strong, a signal that they are strong right now, and it's actually an honest signal in this case. It can't be faked. So that would be the origins of it. That's my claim about the real origins of for why red has to the extent that it has a universal meaning, probably comes from that.
Imai-san?
I don't know. I'm not a psychophysicist, and then it's kind of hard for me to answer the questions. But what I found interesting was red, in both languages, and then probably in most of the languages in the world, the word corresponding to red defer to a relatively narrow space, right? Whereas blue and green defer to relatively broad space, right? And then that seems to be kind of suggested. And, with the fact that children universally acquire red very early, their red is probably one or the two the first or second earliest acquired words was words that they, children start to say, right? And then it's probably red is much more physically or bodily grounded than blue and green. So I don't know if there is a strong contrast between red and green from the beginning. It's red with, with some other colors, right? And then, there is the issue of blue and green, right? Distinction. So English speakers take it for granted that blue and green are distinctive colors, right? And then that reflects RGBY. But there are more languages in the world that do not have special word labels for green, distinct labels for green and blue. And Japanese did not have, like Midori is later coming word, right? They had blue, but Midori came into the lexicon much later, right? And because of that, there is a very much strong confusion between the use of blue and green, Midori and Ao, right? Because Ao gets extended to very typical green color things, right? For example, forest or mountains, we say green, right? And actually green vegetables, we say aona, blue vegetables, right? So something like that confuses children quite seriously, right? So I studied the children's first word, and then we followed the acquisition longitudinally, right? And so they got red and then yellow, fine. And then they say blue very early, but the green came really, really late. And then sometimes they confuse green with purple, right? Or brown. It's because, green, purple, brown are three latest coming words that children get confused, right? So they kind of, randomly map the three that they don't know, right? So given that, I'm not sure if the contrast between red and green is so universal and then, there from the beginning. I mean, it's really an empirical issue. And then we need anthropologists, right, to go to different languages and then study it empirically.
Do you know if there was any kind of study that are like Ariel did, just showing the two patches at the same time, and then ask children of the different culture to rate the similarity across different visual areas?
Oh, well, I don't know.
Okay,
but
that's something that we probably plan to do with Morgan. So good.
Although, yeah, I mean, asking children, if you give children a contrast and then which is more similar than that you can do it. But asking them to judge the sort of, absolute similarity would be hard to get.
Okay.
Yeah, but.
Now, let me share one more thing before, just in that light. So back in the start of it, so when you're looking at, I don't know, but somebody, the distribution. So I'm just showing you a tourniquet. This is really just a bad, early camera from 2005 or something, and trying to show veins. There's some veins going through here. Cameras don't really pick up the veins. And then this is where you've squeezed the blood out of the hand. But the question would be, to the extent that one of the main drivers I'm claiming of the meaning, the ecological meaning of hue is coming from the skin of others, which is one of the most important creatures is, of course, other people, then it could be that the distribution so the question is, what is the distribution in this, or just the distribution of the hues? It could be that when there's red on skin, it tends to be really peaked here, like imagine a histogram that's going around. And then, but when it's in the blue region, there's lots of different kind of blue and green kind of different, it's much more broad over here, and it's really spiked here. One doesn't know, but it could well be the case that even the distribution around that circle in terms of it has some interesting structure, and that we're pretty, so that reds are much easier because we're just used to seeing a really tight bunch of reds, whereas blues and greens tend to be variable, something by the nature of the physiology itself and other complex things about how these colors are shown. When they're chosen to be shown, these are complex mechanisms. These capillaries change and dilate and do all this stuff, and it could be that some of these things just can't happen in this. You can show oxygenated blood, but you can't show deoxygenated blood in the same way you can show whatever. There's some kind of funny constraints. You'd have to really work out the physiology really well and then and measure. And I don't have a good sense of those. But that could be one natural, universal source for some of these differences in the tightness. But again, I don't know how that goes.
Okay, thanks for the great discussion comments. So it's time to close this session. So we will close. There's a question in chat, but please respond to the chat. Okay. So anyhow, yeah, I'll close this session. So thanks for all the participants and the speaker. Thank you so much.
Thank you.
So now, thank you. Now go to the closing comment by Yamada-san, please.
Okay, can you open the slide now? Okay, I'm Makiko Yamada. Let me quickly close the symposium. First of all, we would like to sincerely thank our speakers, Dr. Cohen, Dr. Zeleznikow-Johnston, Dr. Imai, Dr. Changizi for sharing such exciting work, and panelist Dr. Shimojo for giving such thought-provoking discussion. I think 4 hours is not enough, and then we should have another round in near future to go deeper discussion. And I hope many of our audience have also enjoyed the symposium and then would like to get your feedback. Let me paste the link in chat here.
Ariel did that, I think.
Okay, thank you, Ariel.
Yeah.
And I have an additional announcement regarding our future planned events. So could you go to the next slide? Yes, we are planning a symposium on neural and information structure in April or in, is it March or May?
No, May, sorry.
May. And we are also holding a symposium on Qualia structure in Neuro 2021 conference in July. Speakers are us, now Masafumi, myself, and Marieke from University of Western Ontario. These are going to be in English. In Japanese, on March 26th, next week, we are broadcasting Consciousness Radio on YouTube, interviewing Dr. Saigo, who is a mathematician, which would be a very interesting session. Okay. And also, yes, if you want to discuss the issues raised in the symposium, or if you want to know our future activities, please join the Slack. The links are provided here. Okay, in chat also. Okay, lastly, this symposium was successfully held by the helps from Qualia structure staff students, Yota from Monash University, Ryoichi from Kyoto University, Ken from Hokkaido University, Nozomi from Kyoto University, Tomoya from the University of Tokyo. Okay, thank you again for joining us today.
We'll keep you updated through our website and SNS. Okay, see you again.
Thank you very much.
Thank you.
All the speakers and also attendees. Thank you.
Thank you.